Hello there and welcome to Life Beyond Seen, where curiosity gets comfortable and science slows down enough for us to really look at it. Tonight we're turning our attention to a question that sounds simple at first, almost like something a child might ask while staring at a blue globe in a classroom, or peering into an aquarium with their nose pressed to the glass. How many fish are really in the ocean? It sounds tidy. Count the fish, write down the number, and there we are. A nice, clean answer. But the ocean, as it turns out, has never shown much interest in being tidy for our convenience. It is a place of motion, depth, hunger, migration, camouflage, currents, darkness, sunlight, noise, silence, and endless rearranging. Fish are born, eaten, hidden, scattered, netted, tracked, missed, mistaken for one another, and sometimes discovered in places where nobody expected them to be at all. Even the word ocean behaves like a simplification, as if the whole thing were one giant watery room with good lighting and neatly labelled shelves. In truth, it is a restless world layered on top of itself, from warm shallows full of glittering schools to cold depths where creatures drift through black water like thoughts you almost remember and then lose again. So tonight, rather than marching straight toward a neat answer with a clipboard and unreasonable confidence, we're going to do something gentler and, I suspect, far more interesting. We're going to look at what it would even mean to count fish in a place this large, this alive, and this unwilling to cooperate. Because asking how many fish are in the ocean is not quite like asking how many apples are in a basket. It is more like asking how many birds are in the sky if the sky had several floors, some of them invisible, and the birds were allowed to vanish into caves, trenches, coral reefs, kelp forests, river mouths, polar seas, and open water the size of continents. It is, in other words, a counting problem with a flair for drama. Before we get too far into it, I'd love you to settle into the thought with me for a moment. Where are you listening from tonight, and what body of water is nearest to you? Perhaps it is the Atlantic, the Pacific, the Indian Ocean, the Mediterranean, the North Sea, the Baltic, or maybe just a quiet lake, a river, or a stretch of coast you know by smell before you know it by sight. Maybe the nearest water is nowhere near the sea at all, and yet every drop still belongs to the same vast cycle that eventually finds its way back there. It is a strange comfort, really, that even if you are miles from the shore, the ocean still has a way of reaching into your life, through weather, food, trade, memory, and the simple fact that much of the living world depends on what happens beneath those waves. And if you find this little voyage soothing, or interesting, or simply a pleasant place to rest your thoughts for a while, I'd be very glad if you gave the video a like and maybe subscribed. It helps this calm corner of the internet find other listeners who might enjoy a slower walk through big questions. There is no rush, of course. The fish are in no hurry, and for once, perhaps, neither are we. Now, if someone walked up to you tomorrow and said, "Go on then, how many fish are in the ocean?" you might feel an almost immediate urge to guess. People do this very naturally. Our minds like numbers. Even wild guesses can feel satisfying because a number gives the illusion that the world is snapped into focus. 1 million, 10 billion, a trillion, more, less. Something about putting a number on things feels like control. But the more you think about this question, the more it begins to wriggle free of any easy answer. For one thing, fish are not all spread out evenly like raisins in a loaf of bread. They cluster, they migrate, they gather in schools so dense that sonar sees them like clouds, then disperse across huge areas until they are almost impossible to find. Some spend their lives near coastlines, reefs, estuaries and seagrass meadows. Others roam the open ocean in long, patient arcs across enormous distances. Some live near the surface where sunlight reaches, where plankton blooms, where predators hunt with speed and vision. Others descend into dim layers where the light fades to blue and then to nothing. Still others spend their lives in the deep sea, in places that human beings have visited less often than the moon, if we are counting in terms of regular familiarity rather than heroic headlines. And then there is the small matter that the ocean is not empty water with fish sprinkled through it. It is structure. It is habitat. Coral reefs branch and fold into hiding places. Kelp forests create underwater thickets. Continental shelves slope and drop. Seamounts rise from the deep like submerged mountain ranges. Ice edges form shifting boundaries. Nutrients swirl. Temperatures separate populations. Salinity matters. Oxygen matters. Pressure matters. Reproduction matters. Predators matter. Food matters. A fish is never just somewhere. It is somewhere for reasons, and those reasons are always changing. This is where the question begins to reveal its true personality. It is not really asking for a number alone. It is asking how scientists understand abundance in a system too large to empty out, too alive to freeze in place, and too complicated to observe all at once. It is asking how we measure what we cannot fully see. That, to my mind, is where the real interest lies. Not in blurting out a giant figure and feeling pleased with ourselves for 2 seconds, but in learning how knowledge is built from fragments, signals, patterns, and repeated attempts to reduce uncertainty. Because uncertainty, in this case, is not a sign of failure. It is the natural condition of studying a planet-sized moving habitat full of animals that have not agreed to stand in a queue. Imagine trying to count all the trees in a forest, while the forest is spread across several continents. Some of the trees are tiny and hidden under larger ones. Some drift about independently for reasons of their own, and large parts of the forest are underwater and dark. Now add weather, seasons, predation, breeding cycles, fishing fleets, climate shifts, and the fact that some of the trees are actually very good at disappearing into cracks. You would begin, I think, to appreciate why marine scientists do not simply put on a snorkel, glance around and say, yes, seems like about 14,382,119,006. Lovely, let's get lunch. Still, human beings have always tried to make sense of abundance at sea. Long before sonar, satellites, genetic sampling and computer models, coastal communities knew that fish numbers changed. Some years were rich, some years were lean. Some grounds seemed reliable until suddenly they were not. Fish could appear in great silver masses and then fail to return. They could shift routes. They could collapse under pressure. They could rebound when left alone. Entire cultures learned, through labour and error and long memory, that the ocean gives the impression of endlessness right up until it does not. That illusion of endlessness is one of the most important parts of this story. For a long time, many people assumed that the sea was simply too vast for human beings to seriously affect. It seemed inexhaustible. If a place is bigger than your imagination, your instinct is often to treat it as infinite. But infinite is a dangerous word when used carelessly. The ocean is enormous, yes. Its fish populations can be astonishingly large, yes. Yet large and limitless are not the same thing. And history has repeatedly shown that abundance can be damaged, misread, or depleted when people mistake scale for invincibility. So when we ask how many fish are really in the ocean, we are brushing up against several different questions all at once. We are asking how many individual fish exist at any given moment. We are asking how many species there are. We are asking how their numbers vary by place, by depth, by season, and by year. We are asking how much of the total biomass is made up of a few very common species, and how much belongs to countless others that are rarer, smaller, deeper, or harder to sample. We are asking how much we know, how much we estimate, and how much still escapes us. Those are related questions, but they are not interchangeable. And if you mix them up, the ocean will happily let you be wrong for a very long time. There is also something quietly humbling about fish compared with many land animals. On land, people often feel they can picture wildlife with some confidence. Deer in a forest, birds in a park, cows in a field. Even when we do not know exact numbers, we feel oriented. But fish live behind a surface. That matters. The sea conceals by default. You do not casually see its interior the way you see a meadow or a street. Even clear tropical water only reveals a small part of the story, and much of the ocean is not clear tropical water at all. It is cold, rough, dark, deep, turbulent, or remote. We are trying to understand an enormous living system while standing mostly outside it. That barrier between observer and subject has shaped marine science from the beginning. Much of what we know has had to be inferred. A trawl brings up a sample. A sonar return suggests a school. A tagging study reveals a route. A camera records a passing form. A genetic trace in seawater hints that a species is nearby. Catch data from fisheries suggest trends, though they come with biases of their own. Each method opens a window, but none of them gives the whole house. And this is where I think the question becomes unexpectedly comforting, as well as fascinating. There is something reassuring in being reminded that not every worthwhile question yields instantly. Some questions require patience. Some answers come in layers. Some truths can only be approached by combining many imperfect views and admitting that the final picture will still have edges we cannot fill in completely. In a world that often demands immediate certainty, the ocean offers a useful correction. It says, more or less, do your best, bring better tools, try again, stay humble. Of course, fish themselves are not lying awake worrying about our estimates. A sardine is not losing sleep because marine biologists have produced a confidence interval instead of a precise head count. Tuna are not gathering in committee to discuss whether the latest biomass model truly reflects their lived experience. Cod, one suspects, have more immediate concerns, yet for us, counting matters. It matters for ecology because fish are part of food webs that support birds, mammals, invertebrates, reefs, and human communities. It matters for conservation because you cannot protect what you refuse to measure. It matters for fisheries because catching fish without understanding population size is a bit like spending from a bank account while refusing to check the balance and insisting optimism counts as financial planning. And so the question has consequences. If we underestimate fish, we may fail to recognize how rich and varied marine life can be. If we overestimate, we may convince ourselves that extraction is harmless when it's not. If we focus only on commercially valuable species, we may miss the broader structure of ocean life that allows those species to exist in the first place. Even the idea of how many depends on whether we mean numbers of individuals, total weight, breeding adults, juveniles, local populations, global populations, or long-term trends. One species may have countless tiny individuals and very little biomass. Another may have far fewer individuals but a much larger mass. Which seems more abundant depends on the lens you use. That lens matters more than we might think. People often imagine the ocean in dramatic creatures. Sharks, tuna, whales, though of course not fish. Swordfish. Big, fast, charismatic forms slicing through blue water with cinematic confidence. But much of the ocean's fish life is made up of smaller species, many of them little known outside scientific circles or fishing communities. Some rise at night and sink by day. Some shimmer in schools so vast they influence predators, nutrient cycles, and local economies. Some spend their whole existence in places most of us will never see, and yet they may be central to how the ocean functions. If we ask only about the famous fish, we are like tourists visiting a city and concluding that the population consists entirely of people in postcards. And then there is the matter of time itself. Any number we might produce would only ever be a moving answer. Fish populations change by the hour, the tide, the season, the year, and the decade. Spawning events can flood habitats with young. Predation can thin them. Temperature changes can shift ranges. Oxygen loss can squeeze populations into narrower bands. Fishing pressure can reduce numbers. Protected areas can help them recover. El Niño events can rearrange entire marine patterns for a while. A number, if we had one, would not be a monument. It would be a snapshot. Useful, yes. Permanent, no. Perhaps this is why the question lingers so well in the mind. It sounds like arithmetic, but it behaves like ecology. It sounds like a single fact, but it opens into methods, uncertainties, histories, mistakes, technologies, habitats, and scale. It asks us to think not only about fish, but about counting itself, and about the odd human desire to take something vast and living and translate it into digits we can hold in our heads. We will, in time, get to the methods. We will look at what scientists mean when they estimate abundance. We will wander into shallow waters where counting is hard enough, then farther out where the challenge becomes something else entirely. We will consider the species we know, the ones we still miss, and the ways fishing and climate are changing the totals even as we try to measure them. But before any serious attempt at counting can begin, there is a simpler obstacle waiting for us, and it is one the ocean presents without apology. The sea does not stay still long enough to be counted like a cupboard full of plates. It shifts, layers, hides, and carries life with it, and that changes everything. At first glance, the ocean can look almost impressively calm. A flat blue surface, maybe a few ripples, a bit of glittering sunlight, perhaps the kind of view that makes people say things like, "How peaceful," just before the weather changes and a wave reminds them that peace and stillness are not actually the same thing. The sea is very good at looking composed from above. It has had a lot of practice, but underneath that surface, even on the quietest day, the ocean is in constant motion. Water is moving sideways, upward, downward, in circles, in long sweeping currents, in tiny turbulent twists, and in seasonal shifts so large they can reorganize entire ecosystems without asking anyone's permission first. That matters because if you are trying to answer the question of how many fish are really in the ocean, the first obstacle is not the fish themselves. It is the stage they live on. You're not counting animals in a still glass tank. You're counting animals in a moving world made of moving water, shaped by moving temperatures, moving nutrients, moving prey, moving predators, and moving boundaries. The ocean refuses to sit still long enough for a tidy inventory, and frankly, it has no reason to cooperate. On land, even when animals move around, the ground usually stays where you left it. A forest may sway, a herd may migrate, birds may come and go, but the basic architecture remains reasonably dependable. Mountains do not drift off in the night. Meadows do not slide halfway across the country before breakfast. Rivers at least have the decency to stay in channels most of the time. The ocean is different. Its landscape is fluid in the most literal sense. A fish may wake up, if fish can be said to wake up in the way we mean it, in a patch of water that is colder, saltier, richer in plankton, lower in oxygen, or flowing in a different direction than the water it occupied the day before. It may be in the same part of the map, and yet in a very different environment. That shifting environment affects where fish go, how they feed, when they spawn, where their young survive, and whether scientists looking for them are even searching in the right place. If a species tends to gather in cool, nutrient-rich water, and that water shifts because of winds, seasons, or a climate event, the fish often shift with it. If prey concentrates along a front where two water masses meet, predators gather there too. If oxygen levels shrink in deeper layers, fish may be pushed upward into narrower zones. If temperatures rise in one region, a species may expand poleward, retreat from traditional grounds, or alter the timing of its migration. In other words, the fish are moving because the ocean is moving, and the ocean is moving because physics is in charge and does not care whether our spreadsheets were ready. One of the simplest ways to picture this is to imagine the ocean as less of a giant bowl and more of a living transport system. Currents act like conveyor belts, escalators, loops, and one-way lanes. Some are broad and powerful, carrying warm water across whole basins. Others are narrow and fast, hugging coastlines or slipping between islands. Some rise from the deep, bringing nutrients upward in a process called upwelling. Others push surface water away, drawing colder water from below. These movements are not just background details for marine life. They are part of the system that decides where food appears, where larvae drift, where plankton blooms, and where entire populations may thrive or struggle. Consider a young fish just after hatching. For many species, the early stages of life are not especially dignified. There is no tiny underwater school satchel, no stern lecture about responsibility, just a vulnerable little body drifting in a world of currents, predators, and chance. Larval fish often go wherever the water takes them. Some are carried toward productive nursery grounds where food is plentiful and shelter exists. Others are swept into less favourable areas and do not survive. That means ocean motion does not merely affect adult fish distribution. It helps decide which generations grow up at all. And that introduces a problem for counting. If the success of a year's young fish depends partly on how currents happen to move eggs and larvae around, then the abundance of fish in one year may differ sharply from the next, even without dramatic changes in adult breeding numbers. 2 seasons may look similar from a human perspective. Same coastline, same species, same fishing town, same gulls making a nuisance of themselves at the harbour. Yet under the surface, subtle differences in water temperature, current direction, or plankton timing may produce very different outcomes. One year brings strong recruitment, meaning many young survive into the population. Another year produces far fewer. The totals shift, and unless you understand the moving water behind it, the numbers can seem almost moody. The ocean also moves vertically, which tends to receive less everyday attention, because most people are not in the habit of thinking about water having flaws. But it does, in a practical sense. Surface waters mix with wind and weather. Below them sit layers that may be warmer or colder, saltier or fresher, lighter or denser. These layers do not always mix easily. In some places, the ocean becomes strongly stratified, meaning the layers remain relatively distinct. Fish respond to this. Some prefer the brighter upper waters where food is concentrated. Some stay deeper in cooler zones. Some spend daylight in dim depths and rise toward the surface at night to feed. Some shift up and down with seasons, storms, or prey availability, so even if, in some miracle of marine bureaucracy, you could perfectly count all the fish in the top layer of one area, you would still not know how many were below, how many would rise after dark, how many had recently moved through, or how many were about to leave. The ocean is not one habitat. It is many habitats stacked together, some separated by gradients that matter enormously to the organisms living there. Counting fish in it is like trying to count people in a city when you're only allowed to look through one apartment window at a time and the elevators never stop moving. Then there are tides, those regular, dependable, deceptively simple shifts in sea level, driven largely by the moon and the sun. Tides sound straightforward because we learn about them early, and because anything that comes with a printed table starts to feel civilized. High tide at this hour, low tide at that hour, how orderly. But for fish, tides can reshape local conditions constantly. Estuaries fill and drain. Salt water pushes inland and retreats. Currents strengthen through channels. Feeding opportunities change. Access to nursery habitat changes. Small fish move with the cover of water. Predators follow. Entire coastal communities of animals live by these cycles, and any attempt to measure abundance in such places has to reckon with the fact that the same location can function very differently over the course of a day. A shallow bay at low tide and the same bay at high tide are, from a fisher's perspective, not quite the same world. One offers channels, pools, exposed flats, and certain predators. The other opens routes, expands habitat, and changes how food is distributed. If a scientist samples in one tidal state and another scientist samples in another, they may get very different results without either of them being wrong. The sea has simply rearranged the playing field again. Weather adds its own layer of difficulty. Storms stir the surface and sometimes much deeper water too. Winds can push water toward shore or away from it. Rain changes salinity near river mouths. Heat waves warm surface layers. Cold spells cool them. Even short-lived weather events can scatter schools, reduce visibility, alter behaviour, or make certain survey methods less reliable. A patch of water that seemed full of life one week may look sparse the next, not because the ecosystem has collapsed in 6 days, but because fish moved, prey shifted, or rough conditions made them harder to detect. Marine scientists, to their credit, are very aware of this. The ocean, to its credit, remains unimpressed. Large climate patterns complicate things further. El Niño and La Niña, for example, can alter sea temperatures, nutrient supply, productivity, rainfall and currents across vast stretches of the Pacific and beyond. Species that usually thrive in one region may struggle during warm phases. Others may move in. Fisheries that depend on certain populations can swing wildly. The point is not that fish are fickle. It is that the conditions they depend on can be reorganised at continental scales. You're not just counting animals. You're trying to count animals inside a system that periodically reshuffles itself like a dealer who suspects the table has grown too confident. And then there is ice, which many people do not include in their mental picture of the ocean, until the polar regions politely remind them that seawater does in fact have options. Seasonal sea ice changes light levels, habitat access, predator-prey interactions and food availability. As ice forms and retreats, it opens and closes marine spaces. Fish in polar and subpolar seas respond to these changes, as do the organisms they feed on. A survey taken during one period of ice coverage may tell a different story from one taken after retreat. Once again, the ocean refuses to hold still for the head count. Even coastline itself is not as fixed as people imagine. Estuaries shift with sediment, storms, floods and development. Coral reefs grow, bleach, recover or erode. Kelp forests expand and collapse. Seagrass meadows can flourish or vanish. Mangroves advance or retreat. These coastal habitats are crucial for many fish, especially juveniles. A change in habitat structure can alter where fish gather, where they hide, where they find food, and where they can be counted. If you remove a nursery ground, you may not see the consequences immediately in a dramatic cinematic fashion with sad violins and a title card. But over time, the loss may echo through local populations in ways that matter very much. The open ocean, meanwhile, has its own way of being slippery. Out there, away from obvious landmarks, fish often gather around things that are themselves moving or patchy. Plankton blooms, temperature fronts, floating debris, drifting seaweed, prey swarms, upwelling zones. Productivity is not spread out like butter on toast, it is patchy. Life clusters around opportunity. If you sample the wrong patch on the wrong day, you may conclude an area is nearly empty when the action is simply 20 kilometres away attending to a different menu. This patchiness is one reason schools of fish are such a headache for counting. Schooling is a brilliant strategy if you are a fish, and a somewhat irritating one if you are a human trying to estimate abundance. Fish do not spread out in a polite representative sample. They bunch together. Sometimes a lot. A dense school of small pelagic fish such as sardines or anchovies can create enormous local concentrations, while nearby waters appear comparatively barren. If your survey catches the school, your estimate may jump. If you miss it, the area looks far poorer than it really is, and because schools move, split, merge, dive, rise and react to predators or vessels, catching them in statistical terms can be a delicate business. There is also the fact that fish themselves are not passive passengers. Yes, water moves them, but they swim, and often with astonishing purpose. Many species undertake migrations over huge distances, tuna cross ocean basins, salmon move between ocean and river, eels do something so peculiar and ambitious that if they wore tiny backpacks nobody would be surprised. Reef fish may have small home ranges, while others roam widely. Some follow temperature bands, some follow spawning grounds, some track prey, some change depth by day and night in daily rhythms known as diel vertical migrations, which sounds wonderfully formal for something that basically means up at one time, down at another, thank you very much. Those daily shifts are especially important. Many fish and countless other marine organisms rise towards surface waters after dark to feed, then sink deeper during daylight to avoid predators. This means the ocean's biological distribution can change dramatically between day and night. A survey conducted in daylight may encounter fish concentrated at certain depths, while a nighttime survey in the same area finds them higher in the water column or more dispersed. Again, neither picture is wrong. They are simply snapshots of a moving system taken at different times. From a practical point of view, this makes marine surveys a matter of timing as much as place. Scientists have to think not only about where fish are, but when they are there, at what depth, under which conditions, in what season, and behaving in what way. If that sounds exhausting, it probably is. Fish regrettably do not maintain office hours. The ocean's refusal to sit still also creates a problem of boundaries. Humans love boundaries. They make maps easier and management plans look more confident. But fish populations do not always respect the borders we draw. A stock managed by one country may spend part of its life in another's waters or in the high seas. A species that appears abundant in one region may actually be part of a wider population experiencing decline elsewhere. Climate shifts may move long-established ranges across political lines, leading to the sort of international disagreement that starts with scientific reports and ends with people using the phrase historic fishing rights in increasingly tense rooms. From the fish's point of view, all of this must seem terribly administrative. They are following water conditions, prey, breeding cues, and ancient patterns of movement. We are the ones standing on ships with clipboards and jurisdictional headaches. Another reason the ocean resists stillness is that it is linked to the atmosphere in constant exchange. Wind drives surface currents, heat enters and leaves, carbon dioxide dissolves, storms transfer energy, evaporation and rainfall alter salinity. This is not a sealed container. It is part of a planetary system in motion. The atmosphere presses on the sea influences the atmosphere, and fish live in the middle of that conversation whether they like it or not. A marine heat wave can alter local ecosystems. A shift in storm patterns can change mixing and nutrient supply. Long-term warming can alter where species can live comfortably. The result is that what counts as normal in a fish survey may itself be moving over time. That last point is particularly important because people often imagine counting as a path towards certainty, and in many situations it is. Count chairs in a room, count books on a shelf, count biscuits in a tin if you do not live with anyone sneaky. Ocean counting is different. The goal is not to pin life down permanently. It is to estimate abundance within a moving frame and understand how that frame itself affects the estimate. Good marine science does not pretend the water is still. It tries to measure motion, account for it, and work around it. And this is why long-term monitoring matters so much. A single survey can mislead. A series of surveys over years, done carefully and consistently, begins to reveal patterns beneath the motion. Trends emerge. Migration timing becomes clearer. Population changes become distinguishable from temporary shifts in distribution. Scientists learn when certain methods work best and when they are likely to miss things. In a restless system, repetition is not redundancy. It is how the picture sharpens. Still, even the best repeated surveys cannot eliminate the basic truth that the ocean is dynamic in a way that makes clean counting deeply difficult. It is not being difficult out of spite. It is simply being the ocean. Movement is part of how it works. Without currents, mixing, tides, and seasonal shifts, many marine ecosystems would collapse. Nutrients would not circulate in the same way. Larvae would not disperse. Productive zones would not form as they do. Fish abundance itself depends on the same motion that makes abundance hard to measure. The difficulty is built into the vitality. That is perhaps the most important thing to hold on to here. The ocean refuses to sit still because it is alive at scale. It moves because the planet moves. The moon pulls, the wind blows, the sun heats, the earth spins, and gravity keeps working without ever once checking if we're in the middle of a survey. fish live inside that motion. Their numbers are shaped by it. Their locations are shaped by it. Their visibility is shaped by it. To count them honestly, we first have to accept that they inhabit a place where stillness is mostly an illusion seen from shore. And once you accept that, another difficulty arrives, almost immediately in the quiet, mildly annoying way these things often do. Even if the ocean did hold still for us for one miraculous afternoon, we would still need to answer a question that sounds obvious until you try to be precise about it. Before anyone can count fish, estimate fish, model fish, or argue confidently about fish over dinner, we have to decide what exactly we mean by a fish in the first place. That sounds straightforward until you actually try to do it. Fish feels like one of the safest words in the language. Most people hear it and picture something with fins, gills, a tail, and the general courtesy to remain underwater. That seems manageable. A fish is a fish. Job done. Except, of course, that the natural world is rarely interested in being that tidy, and the moment we try to count fish seriously, the word starts to wobble. In everyday life, people use fish quite loosely. We say shellfish, even though mussels, clams and shrimp are not fish at all. We say starfish, though a starfish is an echinoderm and has about as much in common with a cod as a teapot has with a bicycle. We say cuttlefish, which is a cephalopod, meaning it is much more closely related to squid and octopus than to anything with scales and a swim bladder. Language, as always, is very useful right up until it becomes a trap. Science naturally tries to be more precise. In a broad sense, fish are aquatic vertebrates that usually breathe through gills and typically have fins rather than limbs with digits. That gets us closer. It rules out shellfish, starfish, and the entire family of things the English language has casually dragged into the fish bucket out of habit. It also helps separate actual fish from marine mammals like whales and dolphins, which people once insisted on calling fish often enough that history deserves a polite but firm correction. A whale is not a fish. It breathes air, nurses its young, and would probably be offended by the downgrade if it had time for taxonomy. Even with that scientific framing, the category is enormous. Fish are not one neat little group in the way people often imagine. They include jawless fish such as lampreys and hagfish, cartilaginous fish such as sharks, rays and skates, and bony fish, which make up the vast majority of fish species alive today. That last group is huge, and when most people picture a fish, they are usually picturing one of its members, whether it is a tuna, sardine, herring, cod, clownfish, mackerel, or some unimpressed-looking reef resident who appears to have opinions about your diving technique. So already, before we have counted a single animal, we need to decide whether how many fish are really in the ocean includes all of those groups. Scientifically, it should. Sharks are fish. Rays are fish. Eels are fish, no matter how much they look like they were designed by someone trying to make a snake more inconvenient. Lampreys are fish, even if they seem like they belong in a cautionary tale rather than an identification guide. If the question is about fish in the ocean, then the full cast has to be invited, not just the shiny, familiar, commercially valuable members who make it onto dinner plates and wildlife posters. That distinction matters because popular imagination tends to narrow the category without realising it. Say fish, and many people think first of food fish, schooling fish, reef fish, or sporty fish that leap impressively while attached to very expensive equipment. But ocean fish include thousands of species that are small, hidden, deep-living, rarely seen, poorly studied, or of little direct economic interest to humans. A fish in scientific terms does not need to be famous, delicious, photogenic, or large enough to get its own documentary soundtrack. It just needs to be a fish. The trouble becomes even more interesting when we move from common sense to classification. Taxonomy, the science of naming and organising life, is our best attempt at deciding how living things are related. It is useful, rigorous, and occasionally capable of causing mild despair, when new data forces everyone to relearn what belongs where. Fish classification has changed over time as scientists have learned more from anatomy, fossils, and genetics. Species once grouped together are separated. Others once thought distinct turn out to be closely related. In some cases, what looked like one widespread species is actually several cryptic species, nearly identical to the eye but genetically distinct. That matters a great deal if you're trying to count fish accurately, because you cannot count what you have not correctly identified. Imagine trying to count all the people in a city while accidentally lumping together several unrelated neighbourhoods under the same name. Then discovering later that one district was actually 3 different districts, standing very close together in similar jackets. Your original number might still describe a total of people, but it would fail to describe the population properly. The same happens in marine biology. If scientists once treated a set of fish as one species, and later determine it is actually several, then past abundance estimates need to be interpreted carefully. The ocean, meanwhile, continues minding its own business. Then there is the small matter of life stages. Suppose you want to count fish. Do you mean adult fish only? Do you include juveniles? What about larvae? What about eggs? It sounds pedantic until you realise the answer changes the entire scale of the question. Fish begin life in various ways depending on the species. Many lay enormous numbers of eggs into the water or onto substrates. Some guard them, some do not. Some produce live young. Some brood them in odd anatomical arrangements that suggest evolution occasionally likes a bit of theatre. If we are asking how many fish are in the ocean right now, eggs are not usually counted as fish, in the same practical sense as hatched individuals. They are future fish, potential fish, fish in waiting if all goes well, which it often does not. Mortality at the egg and larval stage is famously brutal. The ocean does not offer every hatchling a fair shot and a supportive speech. Larvae, on the other hand, are fish, though they may look nothing like the adults they will become. This is one of the more impolite features of marine life from a counting perspective. Many larval fish are tiny, translucent and bizarrely shaped, as if nature assumed nobody would be looking closely and felt free to experiment. They drift, feed, transform and gradually settle into more recognisable forms. Counting them matters because they represent the future structure of fish populations, yet identifying them is difficult, sampling them is difficult, and linking them back to the right species can be difficult. A larval fish may not look much like the adult field guide version at all, which is inconvenient but entirely on brand for the ocean. So if an estimate focuses on adult biomass, it tells one kind of story. If it includes juveniles, it tells another. If it tries to account for larvae, the numbers swell dramatically and uncertainty goes with them. Each definition serves a purpose, but they are not interchangeable. That is one reason scientists are careful about terms like abundance, biomass, stock size, spawning population, and recruitment. These are not fussy synonyms invented to impress conference rooms. They refer to different pieces of the puzzle. Biomass, for example, means the total mass of living material, not the total number of individual animals. A school of countless tiny anchovies may contain far more individuals than a smaller population of large tuna, while the tuna may outweigh them collectively depending on the scale involved. If someone asks, "How many fish are in the ocean?" a person thinking in terms of biomass and a person thinking in terms of individual animals may produce wildly different mental pictures without either being wrong. One is picturing weight, the other head count. With fish, as with grapes and elephants, the number alone does not tell you everything about abundance. This becomes particularly important because ocean fish vary enormously in size. Some are smaller than your little fingernail at certain life stages, others are larger than a grown human and considerably less interested in your opinions. Some mesopelagic fish, the ones living in the twilight zone of the ocean, are individually tiny but may occur in extraordinary numbers. Their total abundance could be immense, even if each fish is only a small package of muscle and nerves with very reflective sides. So when people imagine counting fish, they often imagine the obvious and miss the statistical weight of the small and numerous. A related complication is habitat. Some fish spend their whole lives in the ocean, others do not. Salmon hatch in fresh water, migrate to sea, then return to rivers to spawn. Eels do the reverse in their own hauntingly committed fashion, growing in fresh or brackish waters before heading to the ocean to reproduce. Some species move between estuaries and coastal seas. Some live near river mouths where salinity changes constantly. So if we are counting fish in the ocean, do we include species whose life cycle depends partly on fresh water, but whose biomass spends substantial time at sea? Usually, yes, if we are interested in ocean fish populations, but again, the wording matters. The ocean is connected to rivers, estuaries, wetlands, and coastlines. Marine life does not stop neatly at the line where a school atlas changed colour. The same issue appears with resident versus transient fish. On a coral reef, some species are basically homebodies. They stay within relatively small territories, defend patches, graze in familiar circuits, and generally behave like local shopkeepers who have seen enough excitement already. Others pass through. Some arrive seasonally to spawn. Some hunt across large ranges. Some juveniles use reefs as nurseries and then leave for deeper or different habitats. If a scientist surveys a reef and counts the fish present that day, are they counting the reef's fish, the region's fish, or merely the fish currently passing through on unrelated business? In practical terms, this affects monitoring and interpretation. The count may be accurate for the moment, but what it means depends on the ecology of the species involved. Another challenge lies in species concepts themselves. Humans like categories because categories are calming. Unfortunately, life has a habit of blurring them. A species is often described as a group of organisms that can interbreed and produce fertile offspring. But even that tidy textbook version has its complications. Some species hybridize. Some populations are geographically separated and diverging. Some lineages look alike but are genetically distinct. Some fish change sex during their lives, which is biologically fascinating and administratively awkward if one prefers simple boxes. Some exist as complexes of near-identical species that only detailed genetic work can untangle. So when counting fish, the question is not only how many are there, but how many kinds are we dealing with, and are we certain where one ends and another begins? Genetics has transformed this field by helping scientists identify cryptic diversity. Fish once assumed to be one broad species sometimes turn out to be multiple distinct lineages. That matters enormously for conservation and management. A large apparent total may mask several smaller populations with different ranges and different vulnerabilities. If you treat them as one big happy unit, you may overfish one component while congratulating yourself on the healthy average. Nature, being under no obligation to reward sloppy aggregation, then reveals the mistake later. There is also the question of whether discovered species are the whole story. They are not. Marine scientists continue to describe new fish species, especially from deep reefs, remote coasts, poorly sampled deep-sea habitats, and regions that have received less scientific attention historically. This does not mean there are millions of giant unknown fish cruising about, dramatically just beyond the edge of our lantern light-like aquatic folklore. It means that biodiversity is still being documented, often in the form of modestly sized, specialized, or hard-to-reach species. If you do not yet know a species exists, you cannot include it properly in global estimates of fish diversity, distribution, or abundance. The unknown is not just philosophical here, it is logistical. And then we come to perhaps the most practical distinction of all. When fisheries scientists talk about fish, they are often not trying to count every fish in the ocean. They are focused on particular stocks, meaning populations of a species managed as a unit for assessment and harvest. A stock is not always identical to a species globally. It might be a regional population with enough separation to behave as its own management entity. So one scientific conversation might be about all fish diversity in the ocean, another about total fish biomass, another about commercially targeted species, another about one spawning stock of Atlantic herring or Pacific sardine. All are legitimate. All sound like counting fish. None mean exactly the same thing. This is where ordinary language starts to become deeply unhelpful. A person might hear, "Scientists estimate fish populations are declining," and assume that means every fish everywhere is disappearing in the same manner. Another might hear that one particular stock has recovered and conclude the ocean is basically fine now. Splendid work, everyone. Let's go celebrate with a grilled fillet. Neither conclusion follows cleanly. Some populations decline, some recover, some shift range, some remain poorly known, some species are heavily exploited, others are barely touched. Fish is too broad a word to carry all of that without support. Even morphology can mislead. There are fish that do not look very fishy in the popular imagination. Seahorses are fish. Pipefish are fish. Frogfish are fish, and seem to have been designed during a particularly playful committee meeting. Sunfish are fish, and one can only admire the confidence with which they move through life, looking like an unfinished idea that became successful anyway. Anglerfish are fish. So are flattened skates, ribbon-like eels, venomous lionfish, transparent larvae, and sleek billfish. The category holds a huge range of body plans, strategies, and habitats. That diversity is part of why counting fish is not like counting marbles in a jar. Even the jar contains several hundred kinds of marbles, some of which are in caves. Some fish are easy to identify visually. Others require expertise. Some can be separated only by fin ray counts, body proportions, otolith structure, colour patterns in life, or genetic markers. Some juvenile stages resemble one species until they resemble another. Some adults change colour by sex, age, mood, breeding condition, or habitat. Reef fish in particular can be unreasonably dramatic about this. A fish guide may show one image, while the fish in front of you appears to have arrived in a different outfit entirely. This is part of the fun if you are snorkeling leisurely. It is less fun if you are trying to turn observations into hard numbers. Sampling gear adds another layer to the meaning of fish. A trawl survey catches the fish vulnerable to that net, at that depth, under those conditions. It misses others. A baited camera records fish attracted or at least tolerant of bait and camera presence. It may miss shy species, hidden species, or tiny species outside the frame. Acoustic surveys detect animals that reflect sound in particular ways, which means some fish show up more clearly than others depending on size, swim bladder presence, behaviour, and distribution. Environmental DNA tells scientists that a species' genetic traces are in the water, but not necessarily how many individuals produced them. In every case, the method shapes what fish means operationally. Scientists know this and calibrate accordingly. The ocean does not make it easy. There is a philosophical edge to all of this, though not the sort that requires dim lighting and overcommitted eyebrow work. Categories are tools. Fish is one of our tools for describing the living world. It is a very good tool, but it is still a human frame applied to a messy, continuous process of evolution and ecology. The animals themselves are not sitting around forming a union called fish and debating membership criteria. They are lineages shaped over immense spans of time. We draw the lines because understanding requires lines, then spend a great deal of effort making those lines more accurate. And accuracy matters because once we say fish, decisions follow. We decide what to survey, what to protect, what to harvest, what to monitor, and what to compare across years or oceans. If a group is ignored because it is small, deep, hard to identify, or not commercially fashionable, our picture of ocean life becomes skewed. If sharks are discussed separately and forgotten in fish totals, we miss part of the story. If deep-sea species are poorly sampled, the apparent centre of abundance shifts toward what we can easily observe. Definition is not a side issue. It is the doorway to measurement. So when we ask what we mean by a fish, the most honest answer is that we mean a vast, diverse set of aquatic vertebrates, whose boundaries are scientifically real but not always simple. whose life stages can look radically different, whose species can be difficult to distinguish, and whose role in counting depends on whether we care about individuals, biomass, breeding populations, commercial stocks, or total biodiversity. That is a lot to ask one small word to handle, but to its credit, fish keeps trying. And perhaps that is the useful shift here. The goal is not to abandon the word because it is imperfect. The goal is to hear it more carefully. Once we do, the ocean starts to look less like a place full of interchangeable silver units and more like an enormous assembly of lineages, strategies, bodies and lives. Some hover near coral heads, some vanish into open blue, some cruise cold shelves, some blink in the dark below sunlight, some are ancient looking, some are bright enough to embarrass fruit, some are counted often, some barely at all. All of them complicate the simple question, which is exactly why the question becomes worth asking properly. Because once we know what we're talking about, or at least once we know how careful we must be with the word, we can finally move toward the places where counting first seems possible. Not the whole ocean at once, certainly not yet, but the coastal shallows, the sunlit waters, the reefs, estuaries, and continental shelves where humans first began to make serious estimates, and where the sea, even there, proved more crowded, patchier, and less cooperative than anyone with a notebook might have hoped. When people first began trying to estimate how many fish lived in the sea, they did not begin in the black depths, far from shore, with robots, sonar, and computers humming confidently in the background. They began where people usually begin with difficult things. They started close to home, in the shallows, in bays, estuaries, reefs, tidal flats, coastal shelves, and sunlit waters where fish could at least sometimes be seen, caught, measured, traded and argued about with a reasonable chance that everyone was talking about the same creature. That made sense. The shallow ocean is where human beings have always met the sea most directly. It is where children wade, where nets are cast, where harbours form, where markets fill, where storms leave their evidence in the rack line, and where for thousands of years people learned that the sea was both generous and unreliable. Coastal communities did not need modern marine biology to know fish numbers changed. They knew from experience. Some mornings the catch was thick and silvery. Some mornings it was sparse enough to sour a whole village before breakfast. Some years the fish seemed to arrive on schedule, as if obedient to old agreements. Other years they did not. People may not have had stock assessment models, but they had memory, labour, and a practical interest in whether the sea was keeping its side of the arrangement. The shallow sea also gave the first real illusion that fish might be countable. Not easy to count, certainly, but countable in principle. In clear water, fish could be seen with the human eye. In tidal pools, they could be trapped by the retreating sea. In estuaries and inlets, they could be sampled with nets. On reefs, they could be observed moving among corals and rocks. On continental shelves, fleets could return with catches large enough to suggest patterns over time. The animals were still mobile, the water was still dynamic, and the habitat was still complex. But compared with the deep ocean, this was the well-lit front room of the marine world. That light matters more than people sometimes realise. Sunlight drives much of the productivity in the upper layers of the sea. Phytoplankton, tiny drifting organisms that perform photosynthesis, form the foundation of many marine food webs. Where sunlight and nutrients meet in useful quantities, life tends to gather. Zooplankton feed on phytoplankton. Small fish feed on plankton. Larger fish feed on smaller fish. Birds, mammals and people join in farther up the chain. The shallow and sunlit sea is not uniformly rich, of course, but it contains many of the places where marine life becomes visible in abundance rather than in theory. Coral reefs are one obvious example. A reef can seem at first like a place where counting fish ought to be almost pleasant. The water may be clear, the fish may be bright. There may be enough beauty in the scene to make a person feel very optimistic about science for several minutes. Then reality returns. Reefs are structurally dense. Fish disappear into crevices, hover under ledges, retreat into branching corals, patrol territories, move in and out of the observer's view, and change activity with time of day. Some are small enough to be missed entirely unless you know what to look for. Others are so quick that you may feel as though they are deliberately testing your professionalism. Counting fish on a reef turns out to be a serious exercise in method rather than a leisurely underwater attendance check. Still, reefs and shallow coasts were among the first places where systematic visual estimates became possible. Divers, snorkelers, and later underwater scientists could conduct transects, moving along a set path and recording fish seen within a defined area. That sounds simple, and in one sense it is. But like many useful methods, it depends on discipline. The observer must identify species quickly and correctly, estimate sizes, avoid double counting, and remain aware that some fish are more visible than others. A bold parrotfish may cruise about in full confidence, while a shy goby remains tucked into the substrate pretending the survey is not happening. If you're not careful, you end up with a very accurate count of conspicuous extroverts and a much weaker grasp of the quieter residents. Even so, these surveys were a major step forward because they moved fish estimation beyond anecdote. Coastal fishers had long known when catches were good or poor, but catch alone is not a direct measure of abundance. If technology improves, fishermen may catch more fish even when populations are shrinking. If weather is bad, the catch may fall even if fish remain plentiful. If effort changes, the meaning of the numbers changes too. A boat with a modest net is not the same as a fleet with engines, refrigeration, electronics, and the collective determination of an industry that has found a profitable shoal. Early scientific estimates in shallow waters helped create something different from catch records. They offered direct observations of fish and habitat. Of course, the first great estimates were not only visual, nets were everywhere in this story. Seine nets, trawls, traps, gill nets and other gear all gave people ways to sample shallow and coastal fish communities. A net haul is useful because it produces bodies rather than impressions. Fish can be identified in the hand, measured, weighed, sexed, aged and compared. Stomachs can be examined. Gonads can be studied to learn about reproduction. Otoliths, the tiny ear bones used to estimate age, can be collected. A net, in other words, turns moving life into data, though usually in a manner the fish would not recommend. Yet nets also have biases, and those biases became clear quite early. Different gear catches different fish. A fine mesh net may collect small juveniles that a larger mesh would miss. A bottom trawl samples fish near the seabed, but not those higher in the water column. Some fish avoid nets, some are caught easily. Some habitats, like rocky reefs or dense vegetation, are difficult to sample with certain gear without tearing the equipment to shreds and causing everyone involved an unnecessary amount of paperwork. So the first serious estimates from shallow waters were never simple counts of everything. They were informed approximations built from selective encounters. Estuaries added another layer of complexity. These places, where rivers meet the sea, are nurseries for many fish species. They are productive, sheltered, rich in food, and often crowded with juvenile stages of fish that later move elsewhere. For scientists, estuaries offered a chance to study young fish in abundance. For counting, however, that abundance came with awkward questions. Were you estimating the local fish community, a temporary nursery pulse, or part of a broader regional population passing through childhood in the shallows before dispersing? The answer depended on the species and the season. A quiet mud flat in spring might contain a swarm of life that would scarcely be recognizable as the same community by late summer. Seasonality became one of the first major lessons of shallow water estimation. Fish abundance near coasts is rarely stable across the year. Spawning seasons bring eggs and larvae. Juveniles recruit into nursery areas. Adults migrate inshore or offshore depending on temperature, food and breeding behaviour. Some species appear in warm months and vanish when cold returns. Others do the opposite. If a scientist sampled a bay in June and again in December, the difference could be dramatic enough to make an unsuspecting outsider think one of the surveys was faulty. But the surveys would simply be revealing that shallow seas are not permanent displays. They are stages used differently over time. This helped push marine science toward repeated surveys rather than single heroic efforts. One estimate, however carefully made, could not define a place. A series of estimates across months and years began to show patterns. Which species were resident, which were seasonal, which habitats acted as nurseries, which areas consistently held dense schools, which populations seemed to be shrinking under heavy fishing pressure. Repetition turned snapshots into a rough-moving picture. That shift was one of the great advances in early marine estimation. It did not solve the problem of counting fish, but it made the problem less foolishly hopeful. The shallow, sunlit zone also gave scientists their first strong encounter with patchiness. Fish were not evenly spread through space. They clustered around reef edges, channels, drop-offs, seagrass meadows, rock piles, oyster beds, and areas where currents concentrated food. This seems obvious now, but obvious things often become obvious only after someone has gone to a great deal of effort to prove them. Patchiness meant that a small number of survey sites could mislead badly if chosen poorly. An apparently rich area might represent a hotspot rather than the norm. A sparse site might sit beside a much denser habitat just out of view. Early estimates improved as scientists learned to stratify surveys by habitat, depth, or region, instead of treating the coastal sea as one uniform bathtub with fish sprinkled in arbitrary places. Seagrass meadows, for example, often proved far more important than casual observers expected. At a glance, they can look like underwater lawns, pleasant enough but not exactly dramatic. To many young fish, however, they are shelter, feeding ground, and refuge. A meadow may contain species that are absent or rare in adjacent bare sand. Mangroves too, where they occur, serve as nurseries and feeding areas. The tangled roots create cover for juveniles, many of which later support offshore fisheries. So an estimate of fish abundance in shallow coastal systems had to include not just the pretty or obvious places, but the muddy, grassy, root-filled habitats that did much of the quiet work of raising the next generation. And then there were the continental shelves, which widened the scale of the problem. These are the submerged edges of continents, often relatively shallow compared with the deep ocean beyond, and many of the world's great fisheries have operated on them. Here, the first great estimates became less about counting visible fish in a reef or estuary, and more about inferring abundance from systematic catches over large areas. Trawl surveys on shelves allowed scientists to sample demersal fish, the species associated with the seabed, in a repeatable way. Stations were set. Gear was standardized as much as possible. Catches were recorded per unit effort. Over time, patterns emerged. This was important because coastal shelves were where the idea of ocean abundance became economically serious. Cod, haddock, plaice, hake, pollock, and many other species supported major fisheries. Governments and fishing industries needed to know whether stocks were healthy, stable, rising, or being fished harder than was wise. The estimates produced were not exact counts of every fish on the shelf. They were indices and models grounded in survey data, but they marked a turning point. Fish abundance was becoming something that could be estimated scientifically, at scales large enough to matter for policy, commerce, and conservation. The results were often sobering. A coastal community may notice when catches decline, but scientific surveys can reveal changes before collapse becomes undeniable. They can show age structures thinning out, breeding adults becoming scarce, juvenile recruitment weakening, or biomass shifting in alarming ways. In some places, the first great estimates of shallow and shellfish populations did not confirm endless bounty. They demonstrated limits. The sea was rich, yes, but not immune. Numbers could fall, entire stocks could be driven down by sustained pressure. The illusion that nearshore abundance was self-renewing no matter what humans did began to crack. There was a practical reason this realization often emerged first in the shallows and on shelves. These were the most heavily used parts of the ocean. They were accessible, they were productive, they were close to ports. Human exploitation reached them early and intensely. If fish numbers were going to tell a cautionary story, these were the places most likely to deliver it. One generation might remember shoals so dense they turned water silver. The next might inherit records, regulations, and a puzzled sense that the stories from older fishers sounded almost mythological. Science stepped into that gap, not to replace memory, but to test it and measure what was changing. Technology naturally improved the estimates. Underwater breathing apparatus expanded direct observation. Better boats extended survey ranges. Echo sounders and early acoustics began to reveal schools in the water column rather than only fish that could be caught or seen. Photography and later video added a record beyond the human eye and memory. But before those tools matured, the shallow sunlit zone had already taught marine science several enduring truths. It taught that visibility is not the same as completeness. Just because you can see part of a fish community does not mean you see most of it. It taught that habitat matters intensely. A sandy bottom, a reef slope, a kelp bed, and an estuary mouth may sit close together on a map and still host strikingly different fish populations. It taught that season matters. A count made at one time of year may not represent the annual picture. It taught that gear shapes knowledge. What you catch is partly a reflection of what your method can catch, and it taught that abundance has structure. Fish are not only many or few, they are distributed by age, size, habitat, season, and behaviour. One of the quiet revolutions in these early estimates was the move from individual curiosity to standardised sampling. A naturalist with sharp eyes could describe a coast beautifully, but one person's impressions are hard to compare over years unless methods are fixed. Standardisation changed that. The same transect, the same trawl duration, the same net type, the same season, the same general location. Repetition under agreed rules allowed trends to become legible. This may sound less glamorous than a diver discovering a brilliant reef at sunrise, and in fairness it is, but it is also how science earns the right to say that numbers are changing rather than merely fluctuating in the imagination. It is worth pausing on how modest these beginnings often were compared with the scale of the question. We started by asking how many fish are really in the ocean. The first serious answers did not emerge as one thunderous figure written across the waves. They came as local estimates: a bay, a reef, a shelf survey, a nursery habitat, a fishery stock, a repeated catch series, a set of transects, a pattern in shallow water. Human knowledge of fish abundance grew coast by coast, habitat by habitat, method by method. The ocean was too large to understand at once, so scientists learned to begin where sunlight helped and where boats could return before supper. That gradual approach also revealed something else. The shallow ocean, though easier to observe than the deep, was already full of uncertainty. If counting fish was difficult here, in clear water, near shore, among known habitats, with repeat access and generations of local experience, what would happen when science tried to push farther out and down? The answer, as it turned out, was that numbers became hazier, assumptions more fragile, and the sea more determined than ever to remind us that visibility is a luxury, not a guarantee. Still, the early work in shallow waters had done something essential. It had shown that abundance could be estimated, that methods mattered, that coastal populations could be tracked, and that fish numbers were neither random nor infinite. The sunlit edge of the ocean gave us the first serious lessons in counting life below the surface. It also gave us a warning. Even where the sea is brightest, fish are not easy to measure, and many of the totals we produce depend on what kind of fish we are looking for, when we look, and what hidden parts of the habitat our methods quietly ignore. And once that becomes clear, the next step is almost unavoidable, because the shallow sea is only the beginning of the ocean's population story. Beyond the bright upper waters lies a region where sunlight weakens, colour drains, and the confidence of neat estimates starts to falter. Out there, in the dim middle depths, numbers do not disappear exactly, but they begin to blur in ways that forced marine science to admit just how little of the living ocean had really been counted at all. Once scientists had learned just how hard it was to estimate fish in the bright, accessible parts of the sea, the next step was not comforting. It was necessary, but not comforting. Because beyond the shallow coastal waters and below the upper sunlit layer lies an enormous part of the ocean where direct visibility drops. Familiar landmarks disappear, and the easy confidence of counting gives way to a different kind of work altogether. This is the region often called the twilight zone, and it is one of the main reasons the question of how many fish are in the ocean remains large, slippery, and far more complicated than most people expect. The twilight zone of the ocean generally refers to the mesopelagic layer, which stretches from roughly 200 meters down to around 1,000 meters, though exact boundaries vary a little depending on conditions and definitions. It begins where sunlight has weakened enough that photosynthesis no longer really powers the system in the same way it does above. There is still some light at the upper part of this layer, but it fades quickly. Colours drain away, shapes become harder to detect. Vision still matters for many animals, but not in the simple, clear waterway people tend to imagine when they picture fish in the sea. It is not full darkness yet, but it is not a place where human eyes are naturally useful either. That alone creates problems for counting. In shallow water, a diver can at least attempt a visual survey. A camera can record recognizable habitats. A fish can be watched interacting with coral, rock, kelp, or sand. In the twilight zone, visibility is limited, the area is vast, the pressure is higher, and many of the animals are small, mobile, and distributed through a three-dimensional volume of water that extends across entire ocean basins. There are no neat underwater lanes marked out for our convenience. There is just layer upon layer of dim water with fish in it, and even that simple statement turns out to need several qualifications. For a long time, human understanding of the twilight zone was thin. Coastal fish and shellfish were familiar because they fed people and lived where people could reach them more easily. Deep-sea creatures captured the imagination because they were strange and dramatic. The mesopelagic layer, by contrast, occupied a sort of awkward middle ground. Too deep for straightforward observation, too broad and difficult for casual sampling, and too full of small species that did not attract the same commercial attention as larger surface fish, or the same theatrical fascination as creatures from the abyss. This does not mean scientists ignored it. It means the layer took longer to become legible. And when it did begin to come into focus, the first clear impression was not neatness, but scale. The twilight zone appears to contain huge numbers of fish, especially small ones. Lanternfish, bristlemouths, hatchetfish, dragonfish, and many other species live there, often in immense abundance. Some are individually tiny, easily overlooked by anyone who prefers their wildlife in a more cinematic size range. But small does not mean unimportant. Small and numerous can add up to a great deal of life, and in the mesopelagic layer that may be exactly what is happening. Take lanternfish, for example. They are among the most abundant vertebrates on Earth, or at least among the strongest candidates for the title depending on how one counts and compares groups. These fish are often only a few centimeters long. They have light-producing organs called photophores, which help with communication, camouflage, and other behaviours that scientists are still studying in detail. They live in large numbers in the twilight zone and are a major food source for larger fish, squid, seabirds, and marine mammals. If you are trying to understand the true number of fish in the ocean, you cannot focus only on tuna, cod, sharks, and reef fish. You have to reckon with the possibility that much of the ocean's fish abundance is tied up in countless small mesopelagic species that spend their lives well below the waters most people ever see. This is where the numbers begin to blur in a serious way. Not because scientists stop trying, but because the methods that work reasonably well in other marine settings begin to run into hard limits. Nets can be used, of course, Midwater trawls have sampled mesopelagic fish for decades. They have revealed species, distributions, size ranges, diets, and abundance patterns. But midwater trawls are not magical. Fish can avoid them. Nets may not open perfectly at the intended depth. Some species are damaged by capture or are hard to identify afterward. Others are missed because they occupy volumes of water that the gear does not sample effectively. A net haul can tell you a great deal, but it still samples only a tiny slice of an enormous moving layer. Fish behaviour makes this worse. Many twilight zone species do not simply drift around waiting to be sampled. They move vertically, sometimes by hundreds of meters over the course of a day. This daily up and down movement, called diel vertical migration, is one of the largest animal migrations on Earth if measured by biomass. At night, many organisms in the mesopelagic rise towards surface waters to feed under the cover of darkness. By day, they sink back into deeper, darker layers where they are less exposed to visual predators. This means the distribution of fish in the twilight zone is not constant even over a 24-hour cycle. Survey at one time and you find one pattern. Survey at another, and the fish are somewhere else, often at different densities and depths. That movement matters enormously. It means that any estimate of abundance has to account not just for location but for timing. A twilight zone fish community is not sitting still in its assigned seats. It is commuting. If you miss the rush hour or catch only one leg of the journey, your understanding of a local abundance may be distorted. This is one reason marine scientists often combine different methods and conduct repeated surveys across times and seasons. Without that repetition, it becomes too easy to mistake movement for absence or concentration for universal density. Acoustics transform this field, but not into something easy. Sonar and echo-sounding devices can detect layers of organisms in the water column by sending sound pulses and measuring the echoes that come back. In many parts of the world, the twilight zone shows up acoustically as a deep scattering layer, a band of life dense enough to reflect sound in a recognizable way. Early researchers were intrigued by this. Some even mistook it for the seabed because it seemed to rise and fall daily. In time, it became clear that the bottom they were seeing was not the ocean floor at all, but masses of living animals moving through the water column. That was a major revelation. It told scientists that the midwater ocean held far more life than had been assumed from sparse net sampling alone. But it also introduced new complications. Acoustics do not count fish individually in the way a person counts sheep through a gate. They detect sound reflections, and those reflections depend on the animal's size, shape, orientation, and anatomy. In particular, many fish reflect sound strongly because of their swim bladders, gas-filled organs that help control buoyancy. Species with swim bladders can create strong acoustic signals. Species without them may be much less conspicuous acoustically. So an acoustic layer is not a perfect census. It is a pattern of echoes that scientists must interpret carefully. This leads to a central problem in twilight zone estimation. If nets may miss fish, and acoustics may overemphasize some species over others, how do you turn either one into a trustworthy estimate of abundance? The answer is that scientists calibrate, compare, model, and revise. They use net samples to identify which species are present, their sizes, and their likely acoustic properties. They compare trawl catches with echo intensity. They build models of how particular groups should reflect sound. They test assumptions. They argue, revise, publish, resample, and then argue again in a more refined way. It is not glamorous, but it is how the blur begins to take shape. Even with these tools, the uncertainty remains substantial. Over the years, some estimates of mesopelagic fish biomass have changed dramatically. In some cases, acoustic data suggested that there might be far more midwater fish than older net-based estimates had implied, perhaps by an order of magnitude or more in certain regions or globally. That is a serious difference. It means the twilight zone may contain a much larger share of ocean fish biomass than scientists once believed. but it also means we're dealing with a part of the ocean where our answers are still somewhat provisional, shaped by method and interpretation. And that is before we consider geography. The twilight zone is not one uniform global layer with a fixed recipe. Conditions vary by region, temperature changes, nutrient supply changes, oxygen levels change, productivity in the waters above changes. Some areas are influenced by upwelling and support-rich food webs. Others are relatively poor in nutrients. Some regions contain oxygen minimum zones, layers where dissolved oxygen is so low that only certain species can tolerate them well. This affects where fish can live, how densely they occur, and how they move vertically through the water column. If oxygen is low in one band, fish may be compressed into narrower zones above or below it. A compressed population can look abundant in one layer and sparse in another, depending on where and how you sample. This spatial complexity is one reason global estimates remain difficult. To know how many fish are in the twilight zone, you would need not only good local surveys, but broad coverage across oceans, seasons, and environmental conditions. You would need to know how abundance changes from productive eastern boundary currents to open ocean gyres, from tropical waters to temperate seas, from subpolar regions to equatorial zones. You would need to know how species composition shifts and how survey methods perform in each setting. This is not impossible in principle, but it is demanding in practice, and the ocean has not become smaller or better behaved while we have been discussing it. The fish themselves, meanwhile, are adapted to this dim world in ways that make them fascinating and difficult in equal measure. Many are small and translucent or darkly pigmented. Some use bioluminescence to hide their silhouette from predators below, matching the faint light from above in a trick called counter-illumination. Some have huge eyes to capture weak light. Some have long teeth, expandable jaws, or stomachs that allow them to make the most of infrequent meals. Some are built for vertical movement over large depth ranges. Many are not species most people will ever hear about, and yet they may form a huge part of the ocean's living machinery. That phrase, machinery, is useful here, but only if used plainly. Mesopelagic fish are not just numerous for the sake of being numerous. They matter because they move carbon, energy, and nutrients through the ocean. When they feed near the surface and return to deeper waters, they help transport carbon downward. When they're eaten by larger predators, they transfer energy upward through food webs. When they excrete waste or die and sink, they contribute to the biological pump that helps move organic matter from surface waters into the deep ocean. So if the twilight zone contains more fish than we once thought, that is not only a counting issue, it changes how we understand global ocean function. This has led to growing interest in whether mesopelagic fish might someday be targeted more heavily by fisheries, and here the uncertainty about numbers becomes more than academic. If we do not know with confidence how much biomass is there, how it varies, how quickly populations turn over, or how central these fish are to carbon cycling and predator diets, then harvesting them at scale becomes a risky proposition. It is one thing to admire a vast hidden resource from afar. It is another to extract it before you understand the system. The ocean has a long history of punishing that kind of confidence. There is also the issue of species richness. The twilight zone contains many species, but not all are equally common, equally widespread, or equally well studied. Some may be abundant across broad regions, others may be localized. Some have distributions linked tightly to water masses or temperature bands. Some may shift ranges with changing climate. If warming oceans alter stratification, productivity, or oxygen levels, twilight zone fish communities may reorganize in ways we're only beginning to understand. So even if a global estimate were firm today, it would still be a moving answer, shaped by environmental change. Sampling this layer is also physically and logistically difficult. Research vessels are expensive. Equipment is specialized. Midwater trawls and acoustic gear require expertise. Collecting, preserving, and identifying fragile deep-living fish is not the sort of work done casually on a pleasant afternoon between coffee and paperwork. Some species come apart easily in nets, others lose key identifying colours or structures after capture. Pressure changes can damage tissues. A haul may contain animals from a broad depth range compressed into one unhappy tangle, and sorting that into clean ecological meaning takes effort and patience. And despite all that effort, there is still the basic problem of scale. The twilight zone covers an immense volume of the global ocean. It is not enough to say that one trawl in one area found many fish, or that one acoustic transect showed a strong scattering layer. The question is how representative those observations are. Marine scientists know this well. That is why large programs compare regions, standardize methods, and build basin-scale or global models where possible. But any honest discussion of ocean fish numbers has to admit that this part of the sea remains one of the major sources of uncertainty. There is something instructive in that uncertainty. People often assume the unknown parts of nature are unknown because no one cared enough to look. In the twilight zone, that is not the case. Scientists have cared quite a lot. The problem is that the ocean is difficult. The layer is vast and the animals do not arrange themselves helpfully. We are not failing to count because we lack curiosity. We are struggling to count because the subject is genuinely hard. Yet even blurred numbers can still teach us important things. We know that the twilight zone holds very large populations of fish. We know these populations play major ecological roles. We know many species migrate vertically each day. We know that abundance patterns vary with productivity, oxygen, region, and depth. We know our methods capture some parts of the story better than others. And we know that older assumptions about where most of the ocean's fish biomass resides may need revision when midwater life is considered properly. That is not a perfect answer, but it is far from ignorance. It also changes the emotional shape of the original question. How many fish are really in the ocean sounds at first as though it should lead to one satisfying number. By the time we reach the twilight zone, the question feels different. It becomes not only about totals, but about visibility, method, and hidden abundance. It forces us to accept that huge portions of ocean life may exist beyond ordinary human notice, not because they are rare, but because they are small, deep, mobile, and distributed through a dim layer that resists easy observation. And that resistance does not end here. Because if the twilight zone blurs the numbers with low light, vertical migrations, and difficult acoustics, the deeper ocean goes further still. Below this middle darkness lies a world with even less direct observation, stranger habitats, and animal communities shaped by pressure, scarcity, and distance from the surface. If the mesopelagic taught scientists that there might be far more fish in the ocean than older estimates had captured, the deeper sea raised another possibility altogether, which is that large parts of marine life had not merely been undercounted, but barely introduced to us properly in the first place. Below the twilight zone, the ocean does something very effective from a counting perspective, which is to become even less cooperative. The last traces of sunlight disappear, pressure climbs steadily, temperature drops and stays low over vast stretches. Food becomes scarcer, habitats more remote, and direct human observation shifts from difficult to expensive, highly planned and never quite frequent enough. This is the deep sea, and for a long time it encouraged a very old assumption, namely that because it was dark, cold, and hard to reach, it must also be mostly empty. That assumption was understandable. It was also wrong in ways that took science quite a while to appreciate properly. The deep sea is not crowded in the way a coral reef is crowded, nor in the way a dense school of sardines turns a patch of water into something like moving metal. You do not descend and find every cubic metre packed with fish, shoulder to shoulder, partly because fish do not have shoulders, and partly because the deep ocean does not work that way. Food is more limited there. Energy arrives more slowly. Much of what sustains deep-sea life begins in the productive upper ocean and sinks as marine snow, a gentle-sounding term for a constant drift of organic particles, waste, remains, and microscopic debris that falls from above. Larger food falls happen too, from dead animals sinking down. But the general rule is that the deeper you go, the farther you are from the main engine of photosynthesis. That tends to reduce the overall density of visible life. And yet, saying the deep sea is not densely packed in the most obvious sense is not the same as saying it contains few fish. What it contains is a great many fish spread across a vast range of habitats, depths, and strategies, plus certain places where abundance gathers more heavily than older assumptions ever allowed. The hidden crowds of the deep sea are not always crowds in the shallow water school-forming sense. Often they are hidden because the habitat is immense, the sampling sparse, and the animals either small, scattered, close to the seafloor, or concentrated in ways we did not recognise until our tools improved. That distinction matters. If you take a quick glance at a deep sea image and see only black water, with the occasional strange face floating through it, you might conclude that fish down there are isolated oddities, each one living a lonely and moderately alarming life in the dark. Some of them probably do live fairly solitary lives, but the deep sea as a whole is not a museum of rare monsters drifting politely far apart for our entertainment. It is a functioning, extensive, inhabited system, and in some regions its fish populations are far richer than early ocean science imagined. The first challenge is deciding what we mean by the deep sea in the first place. In simple terms, people often mean the bathypelagic zone below about 1,000 metres, plus deep benthic habitats along continental slopes, abyssal plains, seamounts, trenches, and other low-light or no-light regions. That already covers an enormous area. It includes the deep open water above the seabed and the seafloor itself, which matters because fish distributions change dramatically depending on whether a species is truly midwater, close to the bottom, or moving between the two. For a long time, much of what scientists knew about deep-sea fish came from trawls, dredges, and the occasional fortunate or unfortunate specimen hauled up by fishing gear working deeper than intended. Those methods were useful, but they were also selective and blunt. Deep trawls sample what enters the net. Fish that live tight against rugged bottom may escape or be missed entirely. Delicate fish can be damaged beyond easy identification. Some species avoid gear. Others may be overrepresented because they are easier to catch. A haul from the deep sea can reveal astonishing forms, but it does not automatically reveal the true structure of the community from which those forms came. That is especially important because the deep sea is not one flat, featureless plain. People often picture it that way because black water and darkness flatten the imagination. In reality, the deep ocean contains slopes, canyons, ridges, seamounts, abyssal plains, trenches, hydrothermal vents, cold seeps, and many kinds of soft and hard bottom habitats. Each of these influences where fish live and in what numbers. A continental slope can support different communities from an abyssal plain. A seamount rising from the deep can concentrate currents, prey, and predators. A canyon can funnel organic material downslope. A vent or seep can host specialized food webs not dependent primarily on sunlight at the surface in the usual way. The deep sea is large enough to contain many neighborhoods, and like all neighborhoods, some are busier than others. Seamounts are a good example of how hidden crowds can form in the deep ocean. These underwater mountains interrupt currents and can create localized areas of productivity or prey concentration. Fish may gather around them in higher densities than in the surrounding deep waters. Orange roughy, alfonsino, certain deep-sea sharks, and many other species are associated with such structures. In some cases, these aggregations became known first through fisheries rather than pure scientific curiosity, which is a mixed blessing. It meant humans found concentrations of fish where the open deep sea had once seemed barren. It also meant those concentrations could be exploited before their biology and vulnerability were properly understood. Orange roughy is a famous cautionary example. It lives in deep waters, often around seamounts, forms aggregations, and turned out to be far more vulnerable than people assumed because it grows slowly, matures late, and can live for many decades. In other words, it is exactly the sort of fish you do not want to discover with a mindset of, well, there seem to be quite a lot here, so this should be fine. Deep-sea fish often live life at a slower pace than their shallower counterparts. Food is limited, temperatures are low, and the economics of survival are different. Some grow slowly and reproduce later. That means abundance can be deceptive. A concentration of adults in one place may look like a robust resource, while actually being a fragile stock that took a very long time to assemble. This is one of the most important lessons from the deep sea. Countability is not just about how many fish exist, it is also about how they are distributed, how quickly they replace themselves, and whether the patch of abundance you found represents genuine resilience or simply a convenient pile of vulnerability. The deep sea has a habit of hiding those distinctions until after damage has begun. The fish themselves are no less varied than those in shallower waters, though they are often less familiar to the public. Grenadiers, sometimes called rat-tails, are common in many deep benthic environments and can be among the dominant fish on slopes and abyssal plains. There are cusk eels, eelpouts, deep-sea cod relatives, slickheads, snailfish, tripod fish, abyssal helisaur species, and a parade of mid-water predators and scavengers whose names alone sound like they deserve dim lighting and a narrator lowering their voice. Some species are broad-ranging and widespread, others are tied tightly to particular depth bands, temperatures, or habitats. Some hover just above the bottom, waiting for food to arrive. Some patrol more actively. Some are built to conserve energy. Some are built to take advantage of rare meals when they appear. The scarcity of food shapes nearly everything in this environment. In the upper ocean, a fish can live in a world where food pulses are frequent and primary production begins nearby. In much of the deep sea, energy arrives second-hand and often in dribs and drabs. That tends to favour efficiency. Mouths may be large relative to body size. Stomachs may stretch. Metabolisms may be lower. Eyes may be reduced in species where light is irrelevant, or enlarged in those using faint bioluminescent cues. Bodies may be soft, because there is no great advantage in carrying around heavy skeletal or muscular architecture when you're not sprinting through bright water, chasing schools in open view. And yet, despite those adaptations to scarcity, the deep sea can still support substantial fish populations over its immense extent. This is where scale returns as the central difficulty. A local density may be low compared with a productive shelf or reef, but the habitat area is vast beyond ordinary intuition. Even modest densities spread across huge slopes and plains can add up to large numbers of fish. The problem is that such numbers are hard to observe directly, because they do not announce themselves in a convenient ribbon near the surface or around a diver's torch. They are distributed through an environment that resists being sampled intensively. Baited camera systems help change that picture. Drop a camera with bait to the deep seabed and fish often appear. Not instantly and not always in the same composition, but often enough to reveal that even seemingly empty bottom can host active communities of scavengers and predators. Grenadiers may arrive, deep-sea sharks may circle in, eels, cusk eels, and other bottom-associated fish may come into view. These systems are useful because they show fish alive in place, behaving naturally enough to tell us something about local communities, rather than only about what survives the trip through a net. But baited cameras have their own biases, of course. Bait attracts the species responsive to bait. It favours scavengers, predators, and species mobile enough to detect and reach the source. It says less about animals uninterested in the offering or unlikely to move toward it. So again, we get a window, not a full map. The history of counting fish in the deep ocean is full of these partial visions. Trawls, cameras, submersibles, remotely operated vehicles, environmental DNA, acoustic tools, fisheries records. Each reveals part of the crowd and misses part of it. The art, or rather the discipline, lies in combining them without pretending they all mean the same thing. Remotely operated vehicles and submersibles gave scientists another important correction. They allowed prolonged direct observation in deep habitats where earlier sampling had relied mostly on capture. This changed how people understood fish distribution. Some species that seemed rare in trawls turned out to be common enough in certain habitats but simply hard to catch. Others behaved differently than expected. Some hovered just above complex terrain where nets were ineffective. Some were associated with biogenic structures, sponge fields, coral gardens, or rocky outcrops that broad gear tended to avoid or damage. Once the cameras went down, parts of the deep sea that had seemed almost vacant began to look less empty and more like neighbourhoods we'd been surveying badly. Deep-water corals are a good case. Cold-water coral habitats on slopes and seamounts can host rich communities of fish and invertebrates. They are not tropical reefs, but they create structure in otherwise less complex settings. Fish gather there for feeding, shelter, or association with prey-rich communities. If you sample only the surrounding soft sediments and miss the structured habitat, you undercount or mischaracterize the local fish population. This is one reason habitat mapping became so important. In the deep sea, knowing where the hard ground, corals, canyons, and seamount features are is not a luxury. It is the difference between looking in the right neighbourhood and staring at an empty parking lot, while concluding the city is underpopulated. Then there is the matter of food falls, which sounds whimsical until you remember that in the deep sea a dead animal sinking from above can be a major event. Carcasses of fish, squid, turtles or marine mammals deliver concentrated pulses of energy to the sea floor. These falls can draw in fish from surrounding areas, creating local crowds that would never be guessed from a single random snapshot elsewhere. A whale fall is the most dramatic example, sustaining rich successional communities for long periods, but even smaller food inputs matter. The deep ocean may appear steady from afar, yet some of its fish communities are organised around irregular opportunities. Count at one moment and one place, and you may see little. Count at another, near a fresh resource pulse, and the scene looks transformed. This patchiness is part of what makes deep-sea abundance so tricky to summarise. The fish are not uniformly sparse, and they are not uniformly clustered. Some are widespread at low density, some occur in habitat-linked concentrations, some aggregate for feeding or spawning, some range broadly over seafloor and water column. The correct answer to how many are there down there is often more than expected but distributed in ways that resist simple averages. Pressure, oddly enough, is less the counting problem than people assume. It is a major biological challenge for the animals, certainly, but not the main intellectual obstacle for us. Fish in the deep sea are adapted to pressure. We are the ones who need expensive equipment and technical expertise to reach them safely. The real counting difficulty lies in remoteness, scale, habitat diversity, and limited survey coverage. You cannot be everywhere. Research cruises are costly. Deep vehicles have limited time and range. Weather interferes. Equipment fails. Identification takes time. Samples are finite. A great deal of the deep ocean still receives only intermittent scientific attention, which means abundance estimates are often assembled from necessarily sparse coverage. This is especially true in the deepest parts of the ocean, such as trenches and huddle zones. Popular imagination loves trenches because they sound dramatic and because they are dramatic. Enormous depth has a way of doing that, but trenches are not the primary habitat for most of the ocean's fish abundance. Many deep-sea fish occur on slopes, plains and midwater layers far shallower than the deepest trench floors. Trenches do contain fish in some cases, including snailfish at astonishing depths, but the deepest hadal environments can become more specialised and less broadly representative than the wider deep ocean. They matter scientifically, but they are not where most people should focus if the question is overall fish numbers. The hidden crowds of the deep are more often spread across the extensive slopes and basins than tucked dramatically into the deepest trench on a poster. Environmental DNA is starting to refine this picture further. By sampling water and detecting genetic traces left behind by organisms, scientists can infer which species are or were recently present. In the deep ocean, where direct observation is difficult, this can be powerful. It can reveal biodiversity missed by traditional surveys and help map distributions across depth gradients. But once again, it does not hand us a clean count of individuals. It tells us about presence, relative signals, and community composition more than exact head counts. So it adds clarity, but not the sort that lets anyone sit back and announce that the ocean has exactly such and such a number of fish and the matter is closed. One of the most useful corrections the deep sea has forced upon marine science is the abandonment of any simple hierarchy of importance based on visibility. Fish near the surface are easier to notice. Fish on reefs are easier to admire. Fish in fisheries are easier to count because markets and regulations keep records. Deep-sea fish, by contrast, spent a long time being underrepresented in both public imagination and scientific totals, not because they were trivial, but because they were out of sight and methodologically awkward. As tools improved, it became increasingly clear that the hidden fish of the deep were not a handful of biological curiosities decorating a mostly empty world. They were and are a major component of marine life. That does not mean the deep sea is overflowing in the same way every habitat overflows. It means that many earlier estimates of ocean fish abundance, especially if anchored too heavily in surface waters, shelves, or commercially obvious species, were bound to miss a substantial part of the story. Some of that missing story lies in the twilight zone's immense midwater populations. Some lies deeper, along bottoms and slopes where fish exist in quieter but still meaningful numbers across huge territories. The ocean's arithmetic changes once you stop counting only where the light helped. There is also a caution hidden here for anyone tempted by the word crowds to imagine inexhaustibility. Hidden crowds can still be vulnerable. Deep-sea fish often have life histories that make recovery slow. Their habitats can be damaged by bottom trawling, mining proposals, and other disturbances. Their populations can appear stable simply because we do not watch them often enough to see changes quickly. In a shallow estuary, a decline may become visible within human routine. In the deep sea, change can remain out of sight for far too long. So the deep ocean enlarges the original question in a very particular way. It tells us that the true number of fish in the ocean is not merely underestimated because the ocean is large. It is underestimated because whole regions of that largeness hide fish in modes we once sampled poorly or barely at all. Darkness misled us. Distance misled us. Rugged terrain, low density, and expensive access misled us. As each of those barriers has softened, the ocean has repeatedly offered the same correction. There is more life here than your first glance suggested. And yet, even after descending into the deep, the problem of abundance is still not only about hidden habitats. Another difficulty remains, one that cuts across shallow waters, midwater layers, and the deep alike. Fish do not simply exist one by one in pleasing isolation. Many of them move in groups, gather in pulses, spread out, tighten up, and form living masses that can turn a survey from neat mathematics into controlled frustration. Because once you know there are hidden crowds, the next challenge is dealing with crowds that move. There is something deeply impressive about a single fish, especially when it is adapted to a place humans rarely see, and manages to get on with its life in conditions that would crush our equipment and our morale at the same time. But if one fish inspires curiosity, many fish together create a different kind of problem altogether. Not for the fish, usually. They seem perfectly content with the arrangement. For us, however, collective behaviour is where counting begins to lose what little composure it had left. Because fish do not only hide in reefs, drift through dim water, or linger near deep slopes. Very often they gather. They school, shoal, swarm, cluster, pulse, migrate in waves, and move in numbers so large that the idea of counting them individually becomes not just impractical, but faintly ridiculous. This is one of the great paradoxes of ocean life. In theory, a fish gathered into a dense mass should be easier to count than a fish spread thinly across a huge area. If the ocean kindly piles its animals together for you, surely that helps. And in some ways, it does. A large school can be detected acoustically. It can be seen from aircraft in shallow, clear water. It can be located by predators, fishers, and scientists alike. Yet the very scale and motion of these aggregations create a new kind of difficulty. A school is not a pile of coins on a table. It is a coordinated, moving, changing body of life. It can turn, compress, stretch, split, merge, dive, rise, accelerate, scatter under attack, reform moments later, and drift out of a survey zone before your equipment has fully decided how impressed it should be. The word school itself is often used loosely, though fish biologists sometimes distinguish between shoaling and schooling. Shoaling refers more generally to fish gathering together for social reasons, feeding, protection, or navigation. Schooling is a tighter, more coordinated form of group movement, where individuals align direction and spacing with striking precision. In everyday speech, most people simply say school, and that is fair enough for most purposes. The important point is that many fish do not live as evenly spaced units across the sea. They live in groups, and those groups can become enormous. Small pelagic fish are the classic example. Sardines, anchovies, herring, mackerel, menhaden, caplin, and similar species often occur in vast schools. These fish are, in many cases, relatively small individually, but there can be extraordinary numbers of them. A school may extend across a large area, or it may compress into a dense, shimmering body that moves with such collective precision, it seems almost less like many animals and more like one flexible organism with thousands or millions of eyes. It is beautiful to watch. It is also a statistical nuisance. The nuisance begins with patchiness. If fish were evenly distributed through the ocean, abundance estimation would still be hard, but at least sampling would be more straightforward. A modest number of representative samples could be scaled up with some confidence. Schooling behaviour ruins that simplicity. One transect may cross a dense aggregation and suggest great abundance. Another, a short distance away, may miss it entirely and suggest relative emptiness. Both observations are real. Neither alone is adequate. The population is there, but it is not distributed in a way that flatters our desire for smooth averages. This is where the difference between local density and regional abundance becomes especially important. A huge school may make one patch of ocean look extravagantly rich, but that does not automatically mean the broader region is packed to the same degree. On the other hand, a survey that misses the school may radically underestimate total abundance. Scientists have to navigate between these two errors. They must avoid being fooled by concentration and avoid being fooled by absence. This sounds simple in the abstract and becomes much less simple once the fish start behaving like fish. Acoustics have been one of the most important tools for dealing with moving millions. Sound travels well in water, and acoustic surveys can detect dense aggregations across large areas far more efficiently than nets alone. When sound pulses encounter fish, especially those with swim bladders, echoes return and can be translated into patterns of density and distribution. For schooling species, this is invaluable. A ship can move through an area and build a picture of where schools lie in the water column, how dense they appear, how deep they are, and how their distribution changes across a survey region. But acoustics, powerful as they are, do not eliminate the problem. A school is not static while the survey vessel passes overhead. Fish may respond to the vessel. They may dive or spread out. Some species avoid ships acoustically or behaviourally, altering the very pattern being measured. Different species reflect sound differently. School shape affects signal intensity. Fish orientation matters. Depth matters. Swim bladder condition matters. And once again, the acoustic return is not a literal count of individual bodies, with names and tiny attendance forms. It is a signal requiring interpretation. This is why acoustic surveys are often combined with net sampling. The acoustics tell scientists where the fish are and how dense the echoes appear. The nets help reveal which species produce those echoes, their size distribution, and other biological details needed to convert sound into abundance estimates. Yet even here, schools resist neatness. A net may pass through the edge of an aggregation, or through a mixed group of species layered close together. Some fish may escape, others may be sampled disproportionately. The ocean does not generally reward attempts to make one method do everything. Schooling also changes with time of day, predator pressure, and feeding behaviour. A tight daytime school may loosen at night when fish spread out to feed. A predator attack may turn a broad school into a compact bait ball, a frantic moving sphere of fish trying not to become lunch. If seabirds, dolphins, tuna or sharks drive a school upward, the fish may compress into surface waters, creating a spectacle that is easy to notice and sometimes easy to catch. But such moments are not the whole story. They are temporary states in a larger behavioural cycle. Count during the drama and you get one impression. Count during the looser feeding period and you get another. The fish population has not necessarily changed. The arrangement has. Spawning aggregations introduce a related but distinct problem. Many fish gather seasonally to reproduce, sometimes in enormous numbers and often in very specific places. Groupers, snappers, cod, herring, and many others may form predictable spawning concentrations tied to lunar cycles, currents, reef promontories, shelf edges, or temperature cues. These aggregations can be biologically vital and statistically misleading in equal measure. On the one hand, they allow scientists to study reproductive populations, estimate spawning stock size, and understand population health. On the other hand, because they concentrate so many adults in one place and time, they can make abundance seem simpler than it is. A spawning site full of fish may represent a healthy population, or it may represent the last crowded meeting point of a stock already diminished over much of its range. One crowded room is not the same as a thriving city. Fishers have long known the importance of such gatherings. Predictable aggregations are efficient to harvest, which is excellent news if one is thinking only about the next catch and less excellent news if one is thinking about long-term resilience. Entire populations can be hit hard when their breeding adults gather densely and predictably. Again, the appearance of abundance can conceal vulnerability. A school of millions feels endless when you stand before it. Biology is under no obligation to agree. There is also the sheer matter of scale. Humans are not especially good at intuitively grasping very large numbers of moving things. We can admire them. We can say that's a lot of fish, which is perfectly true, and often the most emotionally accurate response available. But turning visual mass into quantity is not easy. In shallow, clear waters, aerial surveys or drone footage can sometimes estimate school size by area and density. In aquaculture, where fish occupy pens of known dimensions, machine vision and sampling can help estimate counts. In the open ocean, however, even seeing the whole school is rare. A school can extend beyond the field of view, curve through depth, or fragment into layers. What looks like one aggregation from above may be several groups stacked or braided together. What looks dense at the surface may thin out below. Once again, the ocean turns how many into how exactly are they arranged. Movement makes everything worse and more interesting. A school of herring or sardines is not only numerous, it is coordinated in real time. Individuals respond to the neighbours beside them, to predators approaching, to changes in light, to currents, to hunger, and perhaps to the general social pressure of not wanting to be the one fish who turns left when everyone else is committed to right. Collective movement in fish has been studied as an example of emergent behaviour, where simple local rules produce complex group patterns. Each fish may only need to maintain spacing, alignment, and collision avoidance, yet the whole school behaves with astonishing fluidity. Scientists and mathematicians have modelled this for decades, partly because it is beautiful, and partly because it has implications for ecology, robotics, and our understanding of group behaviour more generally. For counting, however, emergent behaviour means that the shape and density of the group can change quickly. A school is not a fixed geometric object. It is a living cloud with a strong opinion about motion. This matters because survey methods often depend on assumptions about density distribution. If you estimate the size of a school based on acoustic intensity or visible area, you need some sense of how tightly packed the fish are throughout the volume, but density may vary from the core to the edges. The school may flatten, elongate, or turn. Predators may carve through it. Current may stretch it. Another school may merge with it. In the time it takes to measure one piece, the rest may already be becoming something else. Then there are species that do not form the classic silver tornadoes people associate with fish documentaries but still occur in large aggregations. Reef fish may gather in loose feeding groups. Juveniles may cluster in nursery habitats. Deep-sea fish may aggregate around food pulses, seamounts, or current features. Midwater fish can form scattering layers so extensive that they represent moving masses of life on a scale beyond ordinary visual imagination. Not every aggregation looks like a bait ball, but many still create the same problem for abundance estimation. Fish are distributed socially and ecologically, not randomly. One of the more subtle complications is mixed species grouping. The ocean is perfectly happy to ignore our preference for tidy species-specific units. Anchovies and sardines may overlap. Mackerel may associate with herring. Juveniles of one species may occupy similar habitats to another. Predators may shadow prey schools. Acoustic returns may blend signals from multiple fish types. A net haul through a dense area may reveal a mix rather than a pure school. This is not an insurmountable problem, but it means that a school of fish is not always one species behaving in a textbook manner. Sometimes it is a layered social and ecological event with several agendas happening at once. Marine scientists often address this by using multiple sources of information. Acoustic patterns, net composition, environmental conditions, historical knowledge of species behaviour, tagging data, even predator distributions can help interpret what is being seen. But interpretation is the key word there. Large moving aggregations do not surrender exact numbers willingly. They produce evidence, and scientists turn that evidence into estimates with confidence intervals, assumptions, corrections, and occasional healthy skepticism. Tagging has offered another way to understand moving millions, especially for larger schooling or migratory species. Electronic tags can reveal where fish go, how fast they move, what depths they occupy, and how groups shift across seasons and regions. This is especially useful for species like tuna, billfish, and sharks that may travel long distances and gather in productive areas. Tags do not count whole populations directly, but they help solve one major part of the problem by showing how mobile fish really are. A school in one place today may be somewhere very different next week, and management zones based on static maps can look rather naive once the tracks come back. Fishery data also intersect with this problem in complicated ways. Commercial fishing often targets dense schools because dense schools are economical. A captain who can locate a large aggregation of pelagic fish has found not only a biological event but a financial opportunity. As a result, catch per unit effort can remain deceptively high even when populations decline, because fishers continue to locate and exploit the remaining dense schools efficiently. This is sometimes called hyperstability, and it is one of the more treacherous illusions in fisheries science. The catch stays strong not because the stock is healthy everywhere, but because the fish are still concentrated somewhere, and modern technology is very good at finding them. That technological skill has grown dramatically. Sonar, satellite data, bird radar, temperature maps, cooperative fleet knowledge, and increasingly sophisticated electronics all help locate aggregations. Fish, for their part, have not updated their legal counsel. The result is that moving millions can be found and removed at a pace that older generations of fishers could only dream about. This does not mean all fishing is reckless. but it does mean that any serious estimate of fish abundance must account for how school-forming behaviour interacts with human efficiency. A species that clusters tightly can look endlessly available right up until it does not. Climate change adds another moving layer to the story. Schooling fish often track temperature ranges, plankton blooms, oxygen conditions and current systems. As those shift, so do the fish. Entire schools may appear earlier, later, farther north, farther south, or deeper than expected. Migration timing can change. Traditional fishing grounds can become less reliable. Survey designs based on old assumptions may miss newly shifted distributions. This does not only affect the fish themselves, it affects the humans trying to count them, because sampling plans built around past patterns may now be looking in the wrong place at the wrong time with admirable consistency. There is something almost comic in how often this happens. Humans finally develop a decent survey system for a species, congratulate themselves quietly, and then the species changes range because the ocean has warmed or prey fields have shifted. It is not personal. It is simply ecology reminding management that the map is not the territory. All of this is why estimates of highly mobile schooling fish are often expressed with caution. Scientists may be able to assess stock biomass, recruitment, age structure, and trends with considerable sophistication, but exact counts of individuals remain elusive. And frankly, individual counts are often not the most useful measure anyway. For management, what matters more may be spawning biomass, age distribution, reproductive output, and fishing mortality. 1 billion tiny juveniles do not equal 1 billion mature breeding adults in practical terms. Again, the ocean resists our preference for one impressive number to rule them all. Still, the spectacle of moving millions remains central to the public imagination for good reason. It shows something true about ocean abundance. The sea is capable of supporting life in concentrations that feel almost improbable. A bait ball under attack, a wall of herring, a river of sardines, a spawning aggregation filling a reef edge. These are not just dramatic scenes. They are biological facts, and they remind us that fish populations can become enormous under the right conditions. But they also remind us that abundance is often spatially compressed and behaviourally organised. You're not looking at a random sample of all fish everywhere. You're looking at one intense expression of a population in motion. Perhaps that is the heart of the problem. Millions of fish moving together create the impression of certainty because they are visible, concrete, undeniable. There they are. You can see the crowd, but visibility is not the same as representativeness. One great school tells you something important, but not everything. It may reveal productivity, migration, predator-prey dynamics, breeding timing, or local richness. It may also conceal how dispersed the rest of the population is, how many fish remain elsewhere, how quickly they reproduce, or how vulnerable they become when concentrated. So the difficulty of counting fish is not solved by abundance. In some ways, abundance makes it harder, because once fish gather in moving masses, the challenge becomes not only finding them but interpreting what the gathering means. Are we seeing most of the population or only a portion? Are they feeding, migrating, spawning, fleeing, or all of the above in inconvenient sequence? Is this dense because the stock is large or because the habitat has compressed it? Is the school stable or transient? Are we measuring fish or measuring fish behaviour? And that brings us to an important turn in the larger story. Up to this point, we have seen why counting fish is hard because the ocean moves, because habitats differ, because the twilight zone hides abundance, because the deep sea withholds itself, and because fish gather in shifting millions that turn sampling into a game of careful inference. But difficulty is not the same as surrender. Scientists do have methods. Imperfect methods, yes. Biased methods that must be corrected, compared and improved, certainly. Still, they are methods, and they are how the blur becomes something more useful than a guess. Because once you accept that the ocean will not offer a clean head count, the real question becomes how researchers estimate what they cannot fully see. So this is the point where an impatient mind might throw up its hands and say, well then, If the ocean is too big, too dark, too layered, too crowded in some places and too empty looking in others, perhaps the honest answer is that nobody knows anything, and we should all go home. It is a tempting reaction. Also, fortunately, it is wrong. Scientists cannot count every fish directly, but that does not mean they are guessing blindly into the spray. What they do instead is something far more careful and, in its own quiet way, rather ingenious. They estimate what they cannot fully see by combining methods, comparing signals, repeating surveys, and learning how much confidence each tool deserves. That is really the heart of modern fish counting. Nobody is standing at the bow of a ship clicking a handheld counter and trying not to lose track around fish number 43 million. The ocean would win that contest in a morning. Instead, marine science treats fish abundance the way many difficult sciences treat hidden things. It looks for traces, patterns, samples, echoes, movements, and repeatable indicators, then uses those pieces to build a picture strong enough to guide understanding, management, and caution. One of the most familiar methods is the survey net, which sounds almost old-fashioned until you realise how much of marine science still depends on physically catching fish. A net haul provides something wonderfully solid in a field full of uncertainty. It brings back actual animals. They can be counted, identified, measured, weighed, sexed, aged, dissected, compared, and preserved. Scientists can examine stomach contents to learn what the fish have been eating. They can inspect gonads to assess reproductive condition. They can collect otoliths, those tiny ear bones that record age and growth rings a bit like trees, though thankfully much less bark covered. They can separate juveniles from adults, identify mixed catches, and build a biological picture that no remote signal alone can provide. Yet a net only samples part of the world. That is the first rule. Every method sees some fish better than others. A bottom trawl samples fish near the seafloor and misses many that live well above it. A midwater trawl samples the water column and misses much of the benthic community. Mesh size determines what sizes are retained. Some species avoid nets. Some habitats are difficult to trawl without wrecking either the gear or the habitat. A reef is not going to sit politely while you drag a net across it. So net surveys are powerful, but they are never complete. They tell you what was catchable under those conditions, at that place, with that gear, during that time. This is why standardization matters so much. A survey net is most useful not because it catches everything, but because it catches things consistently enough to compare one year to the next. If the same method in the same region starts bringing up fewer cod, or smaller hake, or a thinner spread of age classes, that pattern matters even if the net never captured every last fish in the area. Science often advances not by perfect total vision, but by reliable comparison. It is less about saying, we saw the whole ocean, and more about saying, using the same careful lens, this is changing. Acoustics provide a second major lens, and for many fish they are indispensable. Sound travels efficiently through water, which is very helpful because light often does not. Research vessels equipped with echo sounders send pulses downward and record the echoes that bounce back from fish and other objects in the water column. Dense schools can show up clearly, scattering layers can be mapped. Fish at certain depths can be detected over broad transects that would be impossible to survey visually. If nets give us bodies, acoustics give us structure. They reveal where fish are concentrated and how their distribution changes across space and depth. Still, an echo is not a fish in the hand. Scientists have to interpret what those returns mean. Different animals reflect sound differently. Fish with swim bladders often produce strong signals. Species without them may appear less prominently. The orientation of the fish in the water matters. School density matters. Frequency matters. A return that looks impressive could represent many small fish, fewer larger ones, or a mixed aggregation with properties that change through the water column. This is why acoustics and nets are often used together. The acoustic survey locates and maps the biomass pattern. The nets identify who is actually there. One tool shows the crowd, the other checks the guest list. That combination is one of the clearest examples of how scientists count what they cannot fully see. They rarely rely on one method alone, because one method alone is usually an invitation to be confidently incomplete. Acoustics without biological sampling can misidentify what produced the echoes. Nets without acoustics can miss the broader pattern of where fish were concentrated beyond the net path. Put them together, and the estimate becomes stronger than either on its own. Visual surveys remain important too, especially in shallow coastal systems, reefs, seagrass meadows, and other habitats where divers, snorkelers, remotely operated vehicles or cameras can observe fish directly. Divers may swim transects and record species and numbers within a defined area. Video systems can later be reviewed frame by frame, which has the advantage of letting nobody argue that the fish was definitely there for a second and then vanished just as the observer wrote down something else. Cameras also create records that can be revisited, reanalyzed, and compared by different experts. But direct observation has its own limitations. Fish may hide in crevices, flee from divers, be attracted to lights, remain outside the field of view, or simply occupy the survey area unevenly. Visibility changes with water clarity, current, and time of day. Some species are bold and obvious. Others are masters of being overlooked while technically sitting right there. Anyone who has ever failed to see a camouflaged scorpionfish until it became alarmingly clear they were near one will appreciate this. So visual surveys, like all surveys, become most powerful when repeated carefully and linked to habitat information. Tagging is another key tool, especially for understanding movement, which is essential because fish abundance is always tied to where fish actually are. Some tags are simple physical markers used in mark recapture studies. A fish is caught, tagged, released, and if caught again later, the recapture helps estimate population size or movement patterns. In principle, this is elegantly straightforward. In practice, it depends on enough fish being recaptured, reported, and correctly identified, which means it works better for some species and fisheries than others. Electronic tags take this much further. Acoustic tags can be detected by receiver arrays. Satellite tags can reveal broad migrations for larger species. Archival tags can record depth, temperature, and location estimates over time. These tools show where fish travel, how long they stay in particular regions, when they dive, when they migrate, and how populations overlap. This matters because counting fish in the wrong place is a very efficient way to become impressed by your own mistaken confidence. If a species shifts seasonally, acoustically dense one month and largely absent the next, tagging helps explain that pattern. The fish were not gone from existence, they were gone from your assumptions. Then there are fishery-dependent data, which means information drawn from actual fishing activity, catches, effort, vessel logs, market landings, observer programs, and onboard sampling can all provide insight into abundance trends. This kind of information is often available over long time spans and large areas, which makes it valuable. If a fleet has been landing a species for decades, those records can reveal major changes in size, age structure, location, and relative abundance. But fishery data come with serious warnings. Fishers do not sample the ocean randomly. They go where they expect fish to be, using skill, technology, and accumulated knowledge. That is excellent for catching fish and not ideal for unbiased population estimates. A fleet may continue finding profitable aggregations even while the total population declines, especially in schooling species. Catch can stay deceptively strong because humans become better at locating the remaining fish. That is why fishery-independent surveys, designed for science rather than harvest, are so important. They give a check on the story told by markets and nets aimed at profit. Stock assessment models sit at the centre of much modern fisheries science, and they are perhaps the clearest example of counting through inference. A stock assessment does not pretend to know where every fish is. Instead, it combines many data sources: survey catches, acoustic indices, catch records, age data, Growth rates, maturity schedules, mortality estimates, recruitment patterns, and often environmental information are fed into models that estimate population size, spawning biomass, and likely trends over time. These models are only as good as their assumptions and data, and scientists know that well. They test sensitivity, they compare model structures, they explore uncertainty. They do not simply ask a computer for an answer and nod solemnly while it blinks. In a sense, stock assessment is less like counting coins and more like reconstructing a complicated financial history from receipts, tax forms, and a handful of partial bank statements. You may never see every transaction, but with enough reliable evidence you can estimate the state of the account. That estimate may come with a range rather than a single number, and in science that is a sign of honesty, not weakness. Confidence intervals are what happen when researchers admit the ocean is not a spreadsheet and proceed anyway. Environmental DNA, often called eDNA, is one of the newer methods adding to this toolkit. Fish shed genetic material into the water through skin cells, waste, mucus, and other less glamorous contributions to science. By sampling water and analysing the DNA fragments in it, scientists can detect which species have been present recently. This is especially useful for rare, elusive or deep-living fish that may be missed by traditional methods. It can reveal biodiversity with remarkable sensitivity and help map community composition in places where nets or cameras are limited. What eDNA does not usually provide, at least not yet in a simple way, is a clean count of individuals. A strong DNA signal may suggest many fish, or a few fish shedding a lot, or recent passage through an area, or water movement carrying traces from elsewhere. Interpreting abundance from eDNA is an active area of research. It is promising, but not magical. Like every method before it, it adds a valuable piece rather than a final answer. The ocean remains politely unwilling to be solved by one elegant trick. Satellites, interestingly, help count fish without seeing fish directly. They do not look through the sea in detail because water limits that severely, but they can monitor surface temperature, chlorophyll concentrations, sea level anomalies, ocean colour, fronts, and other indicators of productivity and habitat conditions. Since many fish species track environmental features, satellite data helps scientists predict where fish are likely to gather or move. A productive front, an upwelling zone, a plankton bloom, or a temperature boundary may signal good feeding habitat. This does not count fish outright, but it helps direct surveys and interpret changes. If a species has shifted northward or deeper, environmental data often help explain why. Machine learning and image analysis are beginning to expand what is possible as well. Video footage from reefs, baited cameras, remotely operated vehicles, or fishery monitoring systems can now be processed more quickly and sometimes semi-automatically. Algorithms can assist with species recognition, object detection, and counting in dense footage. This is useful because human experts, though very capable, do eventually get tired if asked to identify the 8,000th fish in slightly murky video from 3 angles and a current. Automated systems are not replacing marine biologists. They are giving them ways to cope with scale, but these systems still require training, validation, and caution. A computer can be fast and wrong with impressive efficiency if not supervised properly. Another important approach is habitat-based modelling. Scientists know that fish are not distributed randomly. They occur in relation to depth, temperature, substrate, oxygen, structure, productivity, and many other factors. So survey data can be linked with environmental and habitat variables to predict where fish are likely to occur across unsampled areas. If trawl stations, acoustic transects, or camera deployments reveal abundance patterns, those can be modelled against habitat characteristics and extended spatially. This is especially useful in large or inaccessible areas where complete coverage would be impossible. Of course, a model is not the same thing as a direct observation. It is a disciplined extrapolation, and scientists know that too. The model must be tested against real data, updated when conditions shift, and treated as a map of probability rather than a divine pronouncement from the sea. Still, these models are crucial because without them, many estimates would remain locked to isolated survey points. The ocean is too large to sample everywhere. Models are how sparse data becomes something broad enough to guide policy. Repeated surveys across time are just as important as broad coverage across space. 1 year of data can mislead. Several years start to reveal trends. Decades reveal something more valuable still. Whether changes are temporary swings, long-term declines, recoveries, regime shifts, or climate-driven reorganizations, long-time series are among the most precious things in marine science. They allow researchers to distinguish seasonal and natural variability from genuine structural change. They also reveal how often the ocean surprises us, which is frequent enough to keep humility in steady employment. Humility, in fact, is one of the hidden methods here. Good fish counting requires it. Scientists must constantly ask what their tools are missing, what assumptions might be wrong, how fish behavior may be altering detection, whether a survey design still fits a changing ocean, whether a model is robust, and whether a reassuring answer may simply be a tidy artifact of incomplete information. There is nothing passive about that caution. It is active skepticism, and it is one reason marine science, despite all the uncertainty, has become far better at estimating abundance than casual observers often realize. A particularly useful concept in all this is the distinction between precision and accuracy. A method can be precise, giving similar results repeatedly, without being accurate if it is consistently biased. A net may sample the same way every year, which is valuable, but if it systematically misses certain fish, the results must be interpreted in that light. Acoustics may map schools beautifully, but species conversion matters. Cameras may see what is in view, but not what stayed hidden. Scientists try to improve both precision and accuracy by comparing methods, calibrating tools, and ground-truthing remote signals with real specimens and observations. This is why marine science often looks less like a single grand invention and more like a coalition of partial witnesses. Nets say one thing, sonar says another, tags trace movement, cameras reveal behavior, DNA hints at hidden presence. Fishers contribute long practical knowledge. Satellites show the conditions shaping habitat. Models weave it together. No one witness sees everything. Together they produce something much more reliable than any could alone. There is also an important shift in what scientists choose to count. In many cases, the goal is not a literal count of individuals, but an estimate of biomass, spawning stock, recruitment strength, or relative abundance over time. That is not an evasion. It is often the more meaningful question. A population of many tiny juveniles is not the same as a strong breeding stock. A fishery may depend less on total bodies than on the number of mature adults and the rate at which young survive to replace them. Ecology may care more about biomass transfer through a food web than about exact head counts. The ocean rarely rewards obsession with the wrong number. Even so, there is something deeply satisfying about how much can be known without seeing everything directly. We do not need to drain the ocean like a bathtub, which would be a terrible idea for reasons both ecological and logistical, to understand fish abundance. We need carefully designed samples, repeated in consistent ways, interpreted with awareness of bias, and strengthened by multiple independent lines of evidence. In that sense, scientists count fish much the way astronomers study distant stars they cannot touch. They do not hold the whole thing in their hands. They read signals, compare patterns, and build models that grow stronger as the evidence converges. And yet, even after all that effort, one more complication remains quietly in the background. Counting fish is hard not only because the fish move and the methods are partial, but because we may not yet know all the fish there are to count. Some species are familiar, well studied and monitored closely. Others are poorly known, recently described, or still waiting in habitats that have not been sampled enough. So even the best counting methods operate in a world where part of the cast is fully documented and part of it is still entering through the side door, unannounced, while the rest of us are pretending the list was complete. And this is where the counting problem becomes even more awkward in a very specific way. It is one thing to say the ocean is large, the fish are mobile, the methods are partial, and the estimates depend on sound, nets, cameras, models, and a great deal of patient correction. That is already plenty to deal with. But there is another difficulty sitting quietly in the middle of it all, and it has a way of making every total feel a little less settled than we might like. Before you can count fish properly, you need to know what kinds of fish are there. Not just in the broad, cheerful sense of, yes, clearly a fish is involved here, but at the level where one species is distinguished from another with enough confidence that your estimate means something real. And that turns out to be a bigger task than people often imagine. Most of us grow up with the feeling that the world's animals have already been found, named, sorted, and placed into reasonably confident lists by people in serious shoes. Lions, whales, salmon, robins, sharks, frogs, and so on. It gives the comforting impression that nature has been inventoried, even if not fully understood. On land, that impression can feel almost plausible, at least for larger animals. In the ocean, however, it is much shakier. Marine science has described a great many fish species, yes, but the ocean still contains species that are poorly known, rarely seen, misidentified, split apart by new genetic work, or still waiting to be described properly at all. So when we ask how many fish are in the ocean, we're doing the arithmetic in a system where part of the cast list remains incomplete. That does not mean scientists are wandering around in total confusion, bumping into unknown fish every Tuesday and having to start over. It means something more modest and more important. The broad outline is known. The fine detail remains unfinished. We know that there are thousands of fish species in the world's oceans, and we know a great deal about many of them. But many is not all, and in a question this large, the difference matters. Part of the issue is simply geography. The ocean contains remote islands, deep reefs, polar margins, trench systems, seamount chains, submarine canyons, and long stretches of continental slope that have not all received the same level of study. Some regions are heavily sampled because they support major fisheries, research institutions, or accessible coastlines. Others remain undersampled for reasons that are entirely practical. They're expensive to reach, politically complicated, difficult to work in, or not seen as a priority until some new evidence suggests they ought to have been all along. Fish do not arrange their ranges according to the Grant calendar, which is inconsiderate of them but very much in character. Then there is habitat complexity. A fish living in open water may be sampled with acoustics or nets more readily than a fish tucked into a coral crevice, hidden in a sponge field, living among mangrove roots, or hugging rugged deep-sea bottom. Small reef fish, cryptic benthic species, and deep-living specialists are especially easy to overlook. Some are tiny, some are nocturnal, some are visible only in particular seasons, depths, or behaviours. Some resemble close relatives so strongly that the human eye, even a trained one, needs help from anatomy or genetics before the truth becomes obvious. It is one thing to miss a large tuna. That is difficult and mildly embarrassing. It is another to miss a small, secretive fish that spends its entire life in a narrow habitat band and has no intention of making itself memorable. This leads us to one of the more interesting ways the species count changes over time. Sometimes scientists discover a species that was previously unknown in the straightforward sense. A specimen is collected or photographed. It does not match existing descriptions. Further study follows. Comparisons are made with museum collections and published records. Anatomy is examined. DNA may be analysed. Eventually, if the evidence holds, a new species is described. That is the classic version of scientific discovery, and it still happens in the ocean more often than many people realise. But there is another route as well, and it may be even more common. A fish long assumed to belong to a known species turns out, on closer inspection, to be several species masquerading under one familiar name. These are often called cryptic species. They may look very similar externally, sometimes almost identical to non-specialists, yet differ genetically, ecologically, behaviourally, or subtly in anatomy. What everyone thought was one widespread fish becomes a cluster of separate lineages, each with its own range and abundance. From a counting perspective, this is rather important. It means yesterday's simple number was not exactly wrong, but it was hiding structure that matters enormously. Imagine being told that one kind of fish is common across half an ocean, only to discover later that it's actually 3 related species, one common, one patchy, and one quite restricted. The total number of fish may not change dramatically overnight, but the meaning of the estimate certainly does. Management changes, conservation concern changes, distribution maps change. The sense of how abundance is spread across the ocean changes. A broad, comfortable average may suddenly conceal a small, vulnerable population that had been riding unnoticed inside the larger label. Genetics has transformed this side of marine science. DNA, barcoding, population genetics, and genomic studies have helped untangle many cases where morphology alone was not enough. Fish that look alike may not be alike. Others that were separated visually may turn out to be variants within one species after all. Larval stages, which can be astonishingly unhelpful in appearance, can now sometimes be linked more confidently to adult species. Tissue samples from museum specimens can be re-examined. Population structure can be studied across whole ocean basins. All of this sharpens the species list, but it also reminds us that the list is not carved into stone tablets in a vault somewhere. It is updated as methods improve. Museum collections deserve more credit in this story than they usually get. Long before genetic tools existed, scientists and explorers were preserving fish specimens from coasts, reefs, rivers, and deep-sea expeditions. These collections are not just dusty rooms full of things in jars, though to be fair, there are jars and a certain amount of dust. They are records of biodiversity through time. A specimen collected decades ago may later turn out to represent an undescribed species. A population once assumed to be part of a common form may be reinterpreted as distinct. Old material can be compared with the new findings to track where species were present historically. In a field where the ocean hides so much, a specimen on a shelf can become an unexpected witness. Photography and video have also expanded the species story, especially in habitats where collecting is difficult or destructive. Deep reefs, for example, have yielded species that were poorly known simply because few people had spent enough time there with the right equipment and enough patience. Remotely operated vehicles have documented fishes at depths that divers cannot routinely reach. High-resolution imagery reveals colour patterns and behaviours that disappear in preserved specimens. In some cases, a species known from only 1 or 2 collected individuals gains context from repeated observations in life. It stops being a mystery in a jar and becomes part of a habitat with actual habits, neighbours, and a schedule. Still, image-based discovery has limits. A photograph can strongly suggest novelty, but formal description usually requires more than a striking picture and a research team saying, "Well, that seems new." Anatomy matters, measurements matter, comparisons matter. Genetics increasingly matters too. The process is careful because it has to be. Naming species is not a game of marine bingo, where the first person to spot an odd fin gets a Latin noun. Scientific description is a way of making biodiversity legible and stable enough that everyone else can build on it. The deep sea, unsurprisingly, contributes heavily to the still missing category. Remote habitats tend to do that. Some deep-sea fish are known from very few specimens. Others are known, but only weakly, with basic taxonomy in place, and much of their ecology still obscure. Some may be more widespread than records suggest simply because nobody has sampled the relevant depth and bottom type often enough. When sampling is sparse, rarity is difficult to interpret. Is the species truly rare or merely rarely encountered by us? The ocean does not answer that immediately. It lets the question sit there until more work is done. Reef systems contribute in a different way. Coral reefs are intensely studied compared with many other marine habitats, but they are also dense with small species, juvenile forms, local endemics, and fishes that hide in narrow microhabitats. Some of the species still being described from reef-associated environments are not giant missing stars of the marine world. They are small, specialized fishes that manage to avoid full scientific recognition because they live in the cracks of a very crowded and visually complicated place. Again, this matters for counting because biodiversity is not only a matter of charismatic large species. A good deal of the ocean's taxonomic richness is built from small forms most people will never see on a calendar. There is also the issue of endemism, where a species is found only in a restricted area. Islands, isolated reefs, enclosed seas, and certain deep features can host endemic fishes that occur nowhere else. These species may have small ranges by global standards, and may therefore be more vulnerable to local change. If such species are undescribed or poorly documented, they do not just slip through the cracks of curiosity, they slip through the cracks of protection. You cannot monitor a species well if you barely know it exists, and you certainly cannot include it properly when speaking confidently about the ocean's total fish diversity. Environmental DNA has begun to help here too. It can reveal genetic signatures of species in waters where traditional surveys are difficult. Sometimes it confirms that a known species is present in an area where it had not been recorded visually. Sometimes it hints that diversity may be richer than the current species list suggests. But hints are not the same as formal species descriptions, and eDNA comes with interpretation challenges. A mysterious signal may indicate an overlooked known species, a poorly sampled population, or something more interesting. It is another light turned into the dark, not a final inventory. And all of this is happening while the ocean itself changes. Species ranges shift. Warm water fishes move poleward in some regions. Cold-adapted distributions contract or reorganize. Some fishes appear in places where they were historically rare or absent. Others decline in areas that once seemed typical for them. When that happens, the line between known species and unexpected appearance can blur. A fish found in a new region may represent range expansion, overlooked historical presence, transport, or misidentification. Sorting that out requires records, specimens, and often genetic confirmation. The ocean, once again, does not simply hand over its paperwork. It is worth noting that the unknowns are not evenly distributed across all fish. Large commercially valuable species are usually better known. Their identities, distributions, and population structures may still contain surprises, but they have been studied more intensively because economies and policies pay attention when dinner is involved. The gaps tend to widen among smaller, deeper, less marketable, or less visible fish. This creates a bias in public understanding. People think we know the fish because we know the fish that matter most in markets, aquariums, and documentaries. But knowing the headline species is not the same as knowing the full marine cast. This is one reason global estimates of marine biodiversity are always offered with some caution. There is a known total, a likely total, and a possible additional margin based on undersampled habitats and taxonomic revision. Fish are better known than many marine invertebrates, to be fair. If you want to feel truly humbled by ocean biodiversity, spend time with the invertebrates and their frankly unreasonable commitment to diversity. But even within fishes, the catalogue is not complete in the absolute sense people often assume, and that incomplete catalogue feeds back into abundance estimates in several ways. First, unknown species cannot be counted directly in global totals of biodiversity. Second, misidentified species can distort abundance patterns, making one widespread fish seem common when several distinct species are actually involved. Third, poorly known species are easier to miss in monitoring and management, which means abundance changes may go unnoticed longer. Fourth, the places where new species are most likely to be found are often exactly the places where counting is already hardest. deep habitats, remote regions, complex reefs, and undersampled margins of the ocean. There is something useful about this uncertainty, though not in the sense of being convenient. It reminds us that scientific knowledge is built, not unveiled all at once. The species we know are the result of centuries of collecting, comparing, naming and revising. The species still missing are not signs that the whole enterprise has failed. They are signs that the ocean remains large enough, detailed enough, and biologically inventive enough that careful work is still uncovering pieces of the picture. In a way, that is exactly what one should expect from the largest living habitat on Earth. It also helps explain why how many fish are in the ocean can never be answered perfectly by one simple number, even in principle. The total is not merely dynamic because fish are born, eaten, harvested and moved by currents. It is also dynamic because our understanding of the species involved keeps improving. The denominator of our ignorance shrinks, but it does not vanish. The labels get better, the boundaries get sharper, the map fills in. Yet there remains, especially in the harder to reach parts of the sea, a margin where the inventory is still being written. This margin is not vast enough to erase everything we know. That point is important. We are not dealing with an ocean where the majority of fish species remain mysterious. Many are well documented. Major families, major fisheries, major regional faunas, and a great deal of ecological structure are known in substantial detail. But the unfinished parts matter disproportionately because they tend to cluster in the exact places where abundance is already hard to estimate, and conservation often lags behind knowledge. And there is one more twist to all of this. Even for the species we know well, the total number of fish is not fixed. Knowing the cast does not tell you how full the theatre is tonight. Species counts and individual abundance are different questions. A reef may host the same broad list of species from one decade to the next, while the numbers within those species change sharply. A fishery species may remain thoroughly known taxonomically, while its biomass rises or falls in response to harvest, climate, prey changes, and habitat shifts. Identification tells us who is in the story. It does not tell us how many remain in each scene, which means that after all this work of defining fish, cataloguing species, splitting cryptic lineages, revising ranges, and admitting that some actors are still entering late, we arrive at another practical truth. The ocean's total is not only hard to calculate because it is incomplete on paper, it is hard because the number itself keeps changing in the real world. Fishing removes fish, climate shifts move them, habitats degrade or recover, some populations rebound under protection, others thin out quietly. The list of species matters, but so does the fact that the size of the living cast is in motion all the time. By this point, the counting problem has already become large enough to deserve a cup of tea and a quiet lie down. The ocean moves, fish move, species are diverse, layered, hidden, mixed, and not always fully catalogued. Scientists rely on careful estimates instead of direct head counts. All of that would be enough to make any number provisional. But there is another reason the total keeps slipping away from neat certainty. And this one is not just about how hard the ocean is to observe. It is about the fact that the number itself does not stay put. Fish are not sitting in the sea waiting to be counted like books on a shelf. They are being born, eaten, maturing, spawning, migrating, and dying all the time. That much is simply life. On top of that natural turnover come two particularly powerful forces that reshape fish numbers in ways humans can feel, measure, and in some cases influence directly. One is fishing, the other is climate change. Together they make the total number of fish in the ocean not just difficult to estimate, but genuinely fluid in the most practical sense. Fishing is the more obvious force because it removes fish directly. Nets, hooks, traps, lines, long lines, trawls, and all the rest are not subtle about their role in the matter. When humans catch fish, there are fewer fish in the water afterward. That is not a controversial insight. But the consequences are more complicated than simple subtraction. Fishing does not remove all fish equally. It tends to target certain species, sizes, ages, places, and seasons. It may remove many adults but leave juveniles. It may target spawning aggregations. It may focus on schooling species. It may pull heavily from coastal shelves, reefs, or upwelling zones while leaving other habitats relatively untouched. So the effect on the ocean's total fish abundance is uneven, structured, and often much more significant than the raw catch figure alone would suggest. A fish population, under healthy conditions, can replace itself. That is why fisheries exist at all. Fish spawn. Young survive at some rate. Some grow to maturity and reproduce in turn. In principle, a human fishery can remove part of that production, while the population remains stable. The key phrase there is under healthy conditions, which has a way of doing far more work than it first appears. Fish populations do not replace themselves automatically at any level of removal. If too many breeding adults are taken, if juveniles have poor survival, if spawning habitat is damaged, if climate disrupts recruitment, or if fishing pressure remains high year after year, then the replacement rate falters and abundance declines. This is why fisheries science is so concerned with spawning biomass. It is not enough to know how many fish there are in total. What matters is how many mature fish remain to produce the next generation. A population can still look busy to a casual observer, and yet be in trouble if too many large breeding adults have been removed. In fact, that is one of the central traps in marine abundance. The sea can still look full while the structure beneath that fullness is thinning out. Smaller fish may remain, young age classes may appear abundant, catches may even stay respectable for a while if fishers are skilled and the species forms dense schools. But a population built increasingly on the young, with too few older breeders, is less secure than it appears. Some species are especially vulnerable to this because of how they live. Fast-growing fish that mature early and produce many offspring can sometimes withstand relatively high fishing pressure if managed carefully. Small pelagic species such as sardines and anchovies can rebound quickly under the right conditions, though they are also famous for natural boom and bust cycles that make management uncomfortable for anyone who likes stable charts. Other species are not nearly so forgiving. Large sharks, many rays, deep-sea fishes like orange roughy, and some groupers, for example, tend to grow slowly, mature late, and reproduce less prolifically. If you fish those heavily, recovery can take a very long time. In some cases, the word recovery starts sounding less like a plan and more like a polite hope. The gear matters too. Bottom trawling can remove fish and disturb seabed habitat at the same time. Gillnets can be highly effective but may create bycatch issues depending on design and management. Long lines target particular species but can also catch non-target animals. Purse seines can take dense schools with remarkable efficiency. None of this makes fishing inherently villainous. Human beings have fished for millennia, and many fisheries are vital to food security and livelihoods. But it does mean that the effect of fishing on the ocean's fish total cannot be discussed as though it were a single neat pressure applied evenly everywhere. It is a set of pressures, each with different consequences depending on species biology, management quality, habitat and scale. Management quality, in fact, is one of the biggest differences between fishing changes the total and fishing steadily dismantles the total while pretending not to. Where scientific assessments are strong, catch limits are enforced, bycatch is reduced, habitats receive some protection, and spawning stocks are monitored, fish populations can stabilise or recover. This has happened in various regions and species. The ocean does not only tell stories of decline, it also tells stories of rebuilding when pressure is reduced and ecosystems are given a chance. But where management is weak, data are poor, enforcement is limited, or short-term economic pressure overrides long-term caution, fish abundance can drop fast enough to shock even people who thought the sea looked endless. Illegal, unreported and unregulated fishing complicates this further, because it removes fish without always appearing clearly in the official record. If catches are not reported properly, scientists and managers are trying to estimate abundance, while part of the subtraction is happening in the dark. That is not ideal, to put it mildly. It means stock assessments may underestimate total removals, making populations seem healthier on paper than they are in the water. Again, the ocean's arithmetic becomes slippery not only because fish move, but because human behaviour does too, sometimes away from the paperwork. There is also bycatch, the fish and other marine animals caught unintentionally while targeting something else. Bycatch matters because it changes total abundance, even when no one set out to remove those particular species. A fishery targeting shrimp may catch juvenile fish. A long line set for tuna may hook sharks. Trawls may take non-target species along with the intended ones. Better gear design, time area closures, and modified fishing practices can reduce bycatch significantly, but the point remains. Fishing alters the ocean's total not only through deliberate harvest, but through collateral removal that can affect species differently, depending on their behaviour and vulnerability. And then there is the food web effect. Removing fish changes more than just the number of that species. It can reshape predator-prey relationships, alter competition, and in some cases shift whole ecosystems. If a major predator is reduced, prey populations may change. If forage fish are heavily fished, seabirds, marine mammals, and larger fish may feel the shortage. If herbivorous reef fish decline, algal growth can change reef balance. This does not mean every fishery causes dramatic ecosystem collapse. It means abundance is interconnected. The total number of fish in the ocean is not a pile of independent units. It is a living network, and removing one piece can echo through others. Fishing then changes the total in direct and indirect ways. It takes fish out, changes population structures, affects reproduction, shifts food webs, and does all of this unevenly across species and regions. But climate change adds a second force that is in some ways even more unsettling, because it does not simply remove fish. It changes the conditions under which fish live, feed, migrate, spawn, and survive. In other words, it changes the rules of the water itself. Ocean warming is the most widely recognised part of this. Fish are ectothermic, meaning their body temperatures and metabolic processes are closely tied to the surrounding environment. If the water warms, their physiology responds. Sometimes that means faster growth or changed activity in the short term, but only within species-specific limits. Beyond those limits, warming can stress fish, alter reproduction, reduce oxygen availability, and shift where populations can live comfortably. A fish adapted to cooler water does not simply shrug and decide to enjoy the tropics. It often moves if it can. If it cannot, it struggles. This is why many fish distributions are shifting poleward or deeper as oceans warm. Species that once occupied certain coastal zones may become less common there and appear more frequently farther north or south, depending on hemisphere, or at greater depths where temperatures remain suitable. That means the total number of fish in one region can decline while another region gains newcomers. From the perspective of a fishing town or a monitoring program built around historical patterns, this can feel like disappearance. From the perspective of the species, it may be a range shift. From the perspective of the global total, the change is more complicated still, because movement interacts with food supply, habitat, and reproductive success. Not every shifted population thrives in its new range. Some simply hang on. Warming also changes timing. Plankton blooms may occur earlier or later. Spawning seasons may shift. Migration windows may move. Juvenile nursery conditions may no longer line up with historical food availability. This matters because fish populations often depend on timing with almost bureaucratic precision. Eggs hatch. Larvae drift. Plankton must be available at the right moment. If warming disrupts that match, recruitment can suffer. One of the quiet ways climate change alters fish abundance is by reducing how many young survive the earlier stages of life, not through spectacle, but through mistimed ecology. Oxygen is another critical part of the story. Warmer water holds less dissolved oxygen than colder water, and many parts of the ocean are already experiencing expanding low oxygen zones. Fish need oxygen, and when it becomes scarce, they either move, become stressed, or die if trapped in bad conditions. In some areas, reduced oxygen compresses fish into narrower bands of habitat nearer the surface, or in particular depth ranges. That can make them more vulnerable to fishing and predation at the same time. It can also make local abundance look strangely high for a while, because many fish are crowded into the remaining livable layer. This is another example of how visible concentration can hide ecological trouble. Ocean acidification, driven by increased carbon dioxide absorption, affects fish less directly than it affects calcifying organisms like corals and some plankton, but it still matters. Fish rely on ecosystems built partly from species that are more sensitive to changing chemistry. Coral reefs, for instance, provide habitat for large numbers of fish. If reefs degrade under the combined pressure of warming, acidification, pollution, and overfishing, the fish communities they support may shrink or simplify. The same logic applies to plankton communities, shell-forming organisms and benthic habitats that fish depend on for food or shelter. Fish abundance is linked to habitat quality, and climate change often reaches fish by first altering the systems beneath their fins. Marine heat waves have added a particularly disruptive element in recent years. These are periods of unusually warm ocean conditions that can last weeks, months, or longer, and trigger rapid ecological shifts. Kelp forests can collapse. Species can move suddenly. Predator-prey patterns can change. Fish communities can reorganize faster than managers or researchers can comfortably track. A survey designed around stable seasonal expectations may suddenly encounter an ecosystem that has changed its mind. In some cases, species historically rare in an area become common for a period, while familiar populations thin out. Again, this affects not only the fish themselves, but the human systems trying to estimate and manage them. There is also the matter of changing currents and stratification. Climate change does not only warm the surface, it can alter mixing, nutrient delivery, and productivity patterns. In some places, stronger stratification can reduce the upward movement of nutrients from deeper waters into the sunlit layer, affecting the food web from the bottom up. If plankton productivity changes, forage fish may change. If forage fish change, predators change. Some regions may become less productive for certain species even if the fish could tolerate the temperature itself. Water conditions are not a single dial marked warm or cold. They are a whole control panel, and climate change is turning several knobs at once. The combined effect of fishing and climate change is especially important because the two pressures do not act separately in tidy sequence. A fish population already reduced by heavy fishing may be less resilient to warming, habitat loss, or poor recruitment. A climate-shifted population may become easier or harder to catch depending on where it moves. A stock may recover under reduced fishing pressure in one decade, only to face new stress from marine heat waves in the next. In other words, the changing total is not shaped by one big force at a time. It is shaped by overlapping pressures, and the balance can differ dramatically from one species or region to another. This is why any simple global statement about fish abundance needs caution. Some populations are down sharply from historical levels. Some are rebuilding. Some are shifting rather than simply shrinking. Some species may benefit regionally from changing conditions for a time, while others lose ground. Small pelagic fish can fluctuate with climate oscillations and fishing pressure in ways that make certainty feel almost rude. Reef fishes may decline where habitat degrades and hold steadier where reefs remain healthier. Deep-sea fishes may be less directly affected by warming at first in some places, but remain highly vulnerable to fishing. The ocean does not deliver one uniform trend line across all fish, and it never did. Yet it would be equally misleading to shrug and say that because the picture is mixed, everything is basically fine. Historical baselines matter here. In many heavily used marine systems, fish abundance has already been shaped for generations by human extraction. The normal people, remember, may itself represent a diminished state compared with older conditions. A coastline that feels familiar and moderately productive today may once have held much larger fish, denser schools, richer reefs, or more complex age structures. This is sometimes called shifting baseline syndrome, and it has a way of making each generation underestimate what has been lost because it treats the recent past as the natural standard. That matters to our original question, because the number of fish in the ocean today is not only hard to know exactly, it is also historically contingent. The total now is not the total a century ago, nor the total before industrial fishing, nor the total in some hypothetical future shaped by better management and stabilised climate. So when people ask how many fish are really in the ocean, there is always an unspoken timestamp attached. How many now, compared with when, under what conditions, relative to which baseline? Without that context, the number floats free of history and loses much of its meaning. The hopeful side of this, and there is one, is that change cuts both ways. Fish abundance can fall, certainly, but under the right conditions it can also recover. Reduced fishing pressure, marine protected areas, bycatch reduction, habitat restoration, and science-based management have all shown the ability to help populations rebound. Some stocks once in serious trouble have improved. Some reef systems show resilience where local stresses are reduced. Some coastal habitats recover when damage is reversed. None of this is quick, and none of it is guaranteed. But the changing total is not always a one-way subtraction. The sea retains, in many places, a capacity for renewal if pressure is moderated in time. That point is worth keeping grounded. Renewal does not mean invulnerability. It means possibility. Fish populations are dynamic by nature. They respond to survival, reproduction, food, habitat, and mortality. Humans now affect all of those more than we once admitted. The ocean's fish total changes because life changes, but also because we've become one of the major forces in the equation. We remove fish, we alter temperature, we alter chemistry, we alter habitats. We then send scientists out with nets, acoustics, tags, and models to work out what remains and how fast it is shifting. There is something very human in that sequence, not always flattering but certainly human. And so by the time we reach this part of the story, the original question sounds different again. How many fish are really in the ocean is no longer just a puzzle about hidden abundance. It is also a question about a moving target shaped by natural cycles, harvest, warming, oxygen, habitat, and time. The total is not waiting behind a curtain to be unveiled. It is changing while we watch, sometimes slowly, sometimes abruptly, always unevenly. Which makes the last step in this quiet journey feel less like a final answer and more like a gentle settling of what we have learned. Because after the methods, the hidden species, the moving schools, the deep-sea crowds, the fishing pressure, and the climate shifts, what remains is not a neat number but a clearer sense of why the number resists neatness at all, and why that mystery, frustrating as it may be for accountants and very determined dinner guests, might be part of what keeps the ocean so compelling in the first place. And so, after all this counting and recounting, sampling and estimating, lowering cameras, dragging nets, reading echoes, tracing migration, splitting species, and trying very hard not to mistake one crowded patch of water for the whole truth, we arrive somewhere both humbling and oddly comforting. The ocean does contain an enormous number of fish. That much is beyond doubt. It contains more fish than any person could ever hope to imagine clearly in one mental picture, and certainly more than any narrator should try to list one by one, unless they have several spare centuries and an unreasonable fondness for administrative work. But the more closely we look, the less this question behaves like arithmetic in the ordinary sense. How many fish are really in the ocean? It sounds, at first, like a question with a grand number waiting patiently at the end of it, like the answer to a quiz hidden in a sealed envelope. A trillion perhaps, several trillion. Some number so large that once you hear it, the mystery gives a polite nod and steps aside. But that is not quite what happens. Instead, the question opens outward. It becomes a question about scale, movement, life cycles, hidden habitats, missed species, difficult methods, changing climates, and the limits of what human beings can confidently know about a world mostly concealed beneath a shifting surface. That does not make the question useless. Quite the opposite. It makes it better. There is something deeply worthwhile about a question that turns out to be larger than its first phrasing suggests. We began with fish, with a count, with what sounded like a problem for a notebook and a patient calculator. Along the way, the ocean replied with currents, reefs, darkness, scattering layers, schools, spawning grounds, seamounts, nurseries, acoustic signals, DNA traces, fishery records, shifting ranges, and species still being sorted into proper focus. The sea did not refuse the question exactly. It simply insisted that any honest answer would need to respect the conditions of the place being asked about. And perhaps that is the real lesson here. The ocean is not difficult merely because it is large. It is difficult because it is alive on a scale that resists simplification. Its fish are not arranged for our convenience. They are arranged by food, by temperature, by oxygen, by predators, by currents, by breeding, by habitat, by chance, and by long evolutionary histories that began very much earlier than our charts and quotas and confidence intervals. We are late arrivals to the scene, standing on boats with excellent equipment and a growing sense that the sea remains under no obligation to become simple just because we have learned how to pronounce biomass. Still, there is progress in all this. We know far more than we once did. We know that shallow seas are only part of the story. We know that the Twilight Zone may hold astonishing amounts of fish life hidden in dim water. We know that the deep ocean, once treated by many as sparse, almost by default, contains substantial communities linked to slopes, seamounts, canyons, and seafloor habitats. We know that fish do not spread themselves evenly like flour across a countertop, but gather in schools, swarms, and moving layers that make any simple count misleading. We know that some species are well studied, while others remain obscure or newly distinguished. We know that fishing removes fish directly, and can reshape populations in quieter ways through altered age structures and food webs. We know that climate change is shifting where fish live and how well they reproduce. And we know that even our best answers must be given with care, because the number itself is always changing. That last part is worth resting with for a moment. The total is not merely hidden, it is moving. It changes from season to season, year to year, decade to decade. Some populations rise, others fall. Some shift northward or deeper. Some recover under better management. Some decline under combined pressure from harvest and warming water. A reef fish community may look much the same to a casual eye, and yet be thinner, younger, or less diverse than it once was. A school may still appear huge while the broader stock behind it has weakened. A once overlooked deep sea layer may turn out to contain far more fish than old estimates allowed. The ocean's arithmetic is not static. It is bookkeeping in a living system, and life is very poor at sitting still for audits. And yet the fact that the answer moves does not make it meaningless. We live quite happily with moving numbers in many parts of life. Population counts change, forest cover changes, bird migrations shift, weather moves, economies rise and fall. We do not throw up our hands and say that because the number changes, measurement is pointless. We measure because change matters. We measure because change reveals whether a system is healthy, pressured, resilient, declining, or recovering. The same is true for the ocean's fish. The point is not to seize the perfect eternal total and place it in a frame. The point is to understand enough of the moving total that our decisions stop pretending the sea is either limitless or fully known. That is, in many ways, where the mystery becomes useful rather than frustrating. mystery is often treated as a problem to be eliminated. But in science, a certain kind of mystery serves another purpose. It reminds us where caution belongs. If you think the ocean is fully counted and fully understood, you are more likely to overreach. If you recognize that large parts of it remain difficult to observe, that many estimates are strong but not absolute, and that hidden abundance can coexist with vulnerability, you begin to think differently. You become more careful with the fish you remove, the habitats you disturb, and the certainty you project. There is a practical ethics in that. Not a grand speech, just a practical one. If you know you are dealing with a system whose total can only be estimated, whose species list is still being refined in places, and whose conditions are changing under human influence, then restraint stops looking like timidity and starts looking like sense. It is one thing to fish, manage, and study the sea. It is another to behave as though everything beyond the hull is a warehouse, with perfect stock reports and no surprises left in it. And surprises do keep arriving. A better acoustic survey revises old biomass assumptions. A genetic analysis splits one species into several. A remotely operated vehicle reveals fish communities in habitats rarely watched before. An environmental shift moves a familiar species beyond its usual range. A stock thought stable begins to show cracks. Another, once badly reduced, shows signs of rebuilding. This does not mean ocean science is unreliable. It means ocean science is alive to revision, which is one of the more trustworthy things a science can be. There is also something pleasantly grounding about the fish themselves in all this. For all our terms and methods, the fish continue their business with complete indifference to whether we have put the correct label on a graph. Lanternfish continue rising at night. Sardines continue gathering when conditions suit them. Reef gobies continue being very small and annoyingly easy to miss. Deep-sea grenadiers continue haunting the slopes in a manner that sounds rude when described plainly, but is for them perfectly normal. Tune across vast distances. Eels carry on with their frankly strange life plans. Cod, groupers, wrasses, snappers, hagfish, sharks, anchovies, dragonfish, sunfish, and countless others go on being fish without once consulting our categories. I think that matters because it gently puts us back in our proper place. We are not outside the story as neutral accountants. We are participants. We fish. We warm the water. We alter habitats. We create protected areas. We fund surveys or neglect them. We build better models. We argue over quotas. We decide which species receive attention and which remain in the shadows longer than they should. The ocean's fish total is not just a fact waiting to be observed. It is a condition shaped partly by us and interpreted entirely by us. That should make us careful, not grand. At the same time, I would not want this question to end in a cloud of solemnity, because there is a lighter side to it too. There is something genuinely delightful in discovering that even in an age of satellites, supercomputers, and highly specific scientific acronyms, a childlike question can still stretch adult knowledge in all directions. How many fish are in the ocean? It sounds like the sort of thing one might ask while drawing waves with a crayon, or staring into a tide pool during a holiday. And yet, the answer touches taxonomy, ecology, fisheries science, acoustics, genetics, remote sensing, oceanography, climate science, and the philosophy of measurement itself. That is one of the pleasures of good questions. They do not always look important at first. Sometimes they wander in wearing simple clothes and then quietly open the whole house. And perhaps it is fitting that the ocean responds in this way. The sea has always had a habit of humbling certainty without entirely defeating curiosity. It lets us know more, but not everything. It rewards patience, but not impatience disguised as confidence. It gives up patterns, but rarely all at once. It is in that sense exactly the kind of place that produces useful science and useful humility at the same time. If we were forced, after all this, to say something plain, it might be this. There are an enormous number of fish in the ocean, likely far more than older generations of science could fully account for, especially once hidden midwater and deep sea communities are included. But there is no single fixed number that can stand forever as the answer, because fish numbers vary through time, differ across species and regions, and depend on methods that estimate rather than directly count the whole living sea. We can know a lot, we do know a lot, but the final total remains a moving estimate, not a neat permanent fact. That may sound less satisfying than a giant headline number, but I suspect it is more honest and in its own way, more interesting. A number can impress you for a moment, an understanding can stay with you longer. Once you know why the count is difficult, the ocean becomes richer, not poorer. You stop seeing fish as a vague mass of underwater silver and start seeing layers of life arranged by habitat, behaviour and hidden structure. You realise that the nearshore waters, the open ocean, the twilight zone and the deep sea are not different versions of the same simple thing. They are connected parts of a vast living system, each contributing to the whole in ways we're still trying to measure properly. And so, as we drift toward the end of this quiet journey, I find myself coming back to where we began, not with a final number, but with the feeling behind the question. There is comfort, I think, in knowing that the world still contains things too large and lively to be pinned down completely. Not because ignorance is comforting in itself, but because partial knowledge, honestly held, has a steadier feel to it than false certainty. The ocean is not a blank. It is not unknowable. It is known in parts, in patterns, in estimates, in repeated surveys, in generations of study, and in answers that improve because people keep looking carefully. And beyond that careful knowing, there remains a margin of mystery that is neither failure nor fantasy. It is simply what happens when one living planet-sized habitat meets one curious species trying to understand it from the shore. So let me ask you, gently, as we come to rest, where are you listening from tonight, and what water is nearest to you now? Perhaps there is a coast not far away, or perhaps the sea is many miles off, and only present in weather, memory, or the salt content of your imagination. Maybe you have stood on a pier and watched bait fish flicker under the surface. Maybe you have seen a market stall full of species whose journeys you never considered. Maybe you have floated above a reef, watched a school turn in one bright motion, and felt for a second that there must be some easy answer hidden in all that movement. Or maybe, like many people, you have simply lived beside oceans in the abstract, knowing they mattered without often thinking about the countless living bodies inside them. If this slow exploration gave you something calming or interesting, or simply a gentler way to think about a very big question, I would be glad if you liked the video, maybe subscribed, and shared this quiet little corner with someone else who might enjoy a slower walk through science. It helps more than you might think, and it lets these careful questions keep finding people who do not mind a long answer to what first sounded like a short one. For now, though, there is no need to solve the sea before sleep. The fish will go on schooling, spawning, rising, sinking, hiding, feeding, drifting, and crossing the dark whether or not we settle the total tonight. Somewhere below a calm surface, a lanternfish is moving upward through dim water. Somewhere on a shelf, a trawl survey is translating a living pattern into numbers. Somewhere on a reef, a small fish is being overlooked by someone with expensive equipment and the best of intentions. Somewhere in the deep, a camera is showing that what once looked empty is not empty at all. And here you are, a little farther along than when we began, with perhaps no final number in hand, but a better sense of why the answer is so large, so lively, and so hard to hold still. The ocean's arithmetic, it turns out, is not the tidy kind written in clean lines on a board. It is the arithmetic of movement, survival, abundance, pressure, chance, and time. Messy, changing, real. And maybe that is exactly as it should be. Sleep well, wherever you are, and whatever the nearest water may be doing tonight.